On behalf of the organizing committee, I would like to welcome you all to this first Human Cell Atlas Latin America Single Cell RNA-seq Data Analysis Workshop. For those who were not here yesterday, we would like to point out that this is the second event in collaboration with the Human Cell Atlas targeting the Latin American region, but it's open to the whole scientific community. So we have a lot of attendees from Europe, also from the US and different parts of Asia. The idea for this workshop came from this first one, which was held last year in September. And several researchers who did not know each other got together virtually at that moment and had the idea of having a second event with a different focus, which would be training. And that's the case of this one. So here I show you guys our organizing committee. We have people from Mexico, Brazil, and Chile, as well as partners from the Human Cell Atlas. Our scientific committee includes a few more people from Mexico, Chile, and Brazil, and also those involved directly with the Human Cell Atlas. For the second week, we have trainers who are based in these 3 countries too, although we are recruiting people from different countries from Latin America. So some of the trainers are based in Brazil, others in Chile and Mexico. And the idea is that there is a project being developed in collaboration with trainers based in these 3 countries. This is the first. We would like to expand this in the near future for other Latin American countries. Here is a slide to acknowledge the institutions and funders that helped us organize this workshop. And we would like also to give a big thanks to those helping us as support for the webinar. Those are our PhD students and postdoctoral fellows that are helping us out. Also the crew from Human Cell Atlas, specifically Luke, Tracy, Samantha, and Christine that was already mentioned, and from the CZI, John Randall and Jonah Kuo. So this is the program for today. We have 2 lectures in the morning, a break of 1 hour when you guys should leave the meeting and come back an hour later. 2 lectures in the afternoon, followed by another break, and then project design discussions for those who are joining us for the second week. For the project design discussion, there is a specific link that is sent to everyone, so it's not the same link that we use for this meeting. So welcome everyone again, and I will introduce our first speaker. So welcome Cheyenne Prabhakar. So Cheyenne obtained a bachelor in technology in electronics engineer from IIT Madras and a PhD in applied physics from Stanford University. He was the sole recipient of the 2001 American Physical Society PhD Thesis Award for beam physics. Following postdoctoral fellowships in mathematics at Stanford and genomics at the Lawrence Beckerley National Laboratory, he joined the Genome Institute of Singapore to start his own research group. His lab uses single-cell analysis, spatial omics, epigenomics, and novel algorithms for basic science and translational studies. Major single-cell research initiatives involving multinational consortia include the Asian Immune Diversity Atlas and an Atlas of Cell States in Colorectal Cancer. He's one of the coordinators of Human Cell Atlas Asia and serves as co-lead of the Human Cell Atlas Genetic Diversity Network. He manages the Genome Institute of Singapore Spatial and Single Cell Genomics platform, heads single cell omics within Singapore's National Precision Medicine Program, and is currently Associate Director, Spatial and Single Cell Systems, and Senior Group Leader in Systems Biology and Data Analytics at the Genome Institute of Singapore. Welcome, Cheyenne. It's an honor to have you here today with us. Thank you.
Thank you very much, Patricia. Thrilled to be here, and exciting times. Being part of very great program you guys have put together. I enjoyed some of the talks yesterday as well. So let me share my slides here. Okay, I presume you can see this now. So I'm going to be talking today a little bit about what's happening in Human Cell Atlas Asia, which I think can be useful because it's a parallel effort, I would say, to perhaps what people in Latin America, Human Cell Atlas Latin America, are doing or planning to do, which is to develop regional consortia and develop science, single cell based science or Human Cell Atlas relevant science that's also relevant for that regional consortium for that part of the world. So let me start with that. And we were inspired by the basic goal of the Human Cell Atlas, which is to create a comprehensive reference map of the types and properties of human cells, okay? And I'll emphasize the word comprehensive reference map. Now, you can be comprehensive in many ways, right? And by default, everyone thinks comprehensive means, okay, so let's try to cover all the possible cell types in the human body. And that is certainly one way you can be comprehensive. But then you also want to be comprehensive in a few different ways. And one of them is the genetic comprehensiveness, okay? So here you can see some other consortia, some other genetics-based consortia or international efforts. One is, say, GWAS, Genome-Wide Association Studies. The other is NOMAD, genome and exome sequencing. And another is GTEx. That's a consortium that's aimed at identifying the genetics of gene expression in human populations. So all of these consortia, they wanted to be comprehensive in many ways, just like the Human Cell Atlas. And I would say they've made fantastic efforts, and these are amazing resources for science worldwide, okay? But there's one challenge that they all faced, and a challenge that the Human Cell Atlas also faces, and that is being genetically comprehensive, or comprehensive in terms of primarily ancestry and geography, okay? I think they're not doing so badly in terms of gender diversity, but in terms of ancestry diversity and geography diversity, it's difficult, simply because of the geographic locations in which this research was carried out. So you can see here that GWAS studies, they're primarily red, okay? Red means European ancestry. Then if you look at NOMAD on the right-hand side, the red boxes indicate populations of European ancestry. And you can see that more than half of NOMAD is of European ancestry. Then you look at GTEx, this blue majority of the pie here is again European ancestry, okay? So it's not from lack of trying. They all wanted to prioritize diverse ancestries and diverse populations, but then, and they made substantial headway, right? But we need to do better. And the Human Cell Atlas also needs to do better. Why is that important? So moving from genetics to disease epidemiology. Okay. So on the left, you see here, this is coronary artery disease mortality rate as a function of, on the x-axis, total cholesterol, okay, your serum cholesterol. Now you can see here that if you live in Japan, your coronary artery disease mortality rate is pretty low, almost regardless of your serum cholesterol. If you're an Asian Indian like me, and particularly male, your risk of dying from CAD is pretty high, and it gets even higher as your cholesterol level goes up. So that's startling, right? So ancestry and or geography matters quite dramatically and really, determines your risk of living or dying, right? Given the same serum cholesterol. So that's a dramatic example of how diseases care about your ancestry and your geography. Then on the right-hand side, you'll see prevalence of diabetes. Again, quite substantially different. even age control is quite substantially different in different populations. And these are just two examples, right? For almost any disease, you'll find that there's some difference in the biology, some difference in the epidemiology. If you go to a different part of the world, or you stay in the same part of the world and you look at a different ancestry. And so that is strongly motivating, right? So if you want to do global genomic medicine, or you want to do precision medicine worldwide, you want to do your genomics in a global fashion as well, right? And that is a key message for us. And so that's what inspired us to start the IDA flagship project within Human Cell Atlas Asia. And Human Cell Atlas Asia, hopefully you'll find it relevant as kind of you can say a sister organization to Human Cell Atlas Latin America, and we have similar goals. So we wanted to expand the diversity of the Human Cell Atlas in a very specific manner by expanding the pool of Asian ancestry samples in the atlas, and also furthering the equity and diversity goals of the Human Cell Atlas. Okay, and so very thankfully we were founded by CZI to do a pilot scale project in this direction. And I've listed the names of my co-investigators in this IDA project. There's Jae-shin leading the Riken team in Japan, Wongyang Park leading the Samsung Genome Institute team in South Korea, Partha Majumdar leading the Indian team in NIBMG, and Varadom Charan Sawan leading the Maidol University team in Thailand. Right? So, and myself coordinating in Singapore, along with my collaborators, John Chambers and Don Chong. So you can see 5 different countries in Asia, and actually 6 ethnicities, because Singapore is a multi-ethnic country. So you have Malay ethnicity, Indian ethnicity, Chinese ethnicity, all in one country at reasonable proportions. So the goal of IDA was to create a single cell atlas of Asian immune cell states, and then examine differences both within a population in Asia and across Asian populations. And then phase 2, so this is all healthy samples, and phase 2 we would compare to Asian disease cohorts. Okay, and we're looking at immune diversity primarily starting with PBMCs, peripheral blood mononuclear cells, from healthy subjects, because that's the most stable and robust place you can start, and that's a great reference data set for comparing to various immune-related diseases. Okay. So that's a broad scope of IDA. And you can see in the background, the green countries on this global map are Asian countries. So Asia covers a very large fraction of the globe, a very large proportion of the world's population, tremendously diverse in terms of genetics and in terms of geography and in terms of environment, right? So I think we're just scratching the surface of Asian diversity here with this initial IDA pilot project, and we need to do much more, but one has to start somewhere. And I should say, be happy to speak to anyone else building regional consortia of this sort if I can share our experiences, if that might help. It's really exciting and wonderful to connect with teams all over the region and realize that we have very similar goals, right? But it's also logistically very, very challenging. And I think that's where our experiences might help. So this is the IDA team. I mentioned the country leads on the left, and we have multiple additional co-investigators, Ian Tan, John Chambers, Don Chong in Singapore, Chung Cha Hon, and Ario Kawakami in Riken, Japan, Arinda Maitra in NIBMG with Partha, and Ankita Chatterjee as well, working with Partha, and then Ponpan in Maidol University alongside Varadam. And this is just the initial team that's been funded by CZI, but actually additional teams are joining IDA as well. So this is not the sum total of IDA. So here's some pilot data, and for this I had to thank Deepa Rajagopalan and Kuei Lin from my team in Singapore, who generated the Singapore data, and Kuei analyzed this pilot data from multiple countries. So this is a very early pilot data set from 3 countries, Japan, South Korea, Singapore, 43,000 PBMCs. And you can see here the classic PBMC clustering in gene expression space. Each color indicates one cell type. And this is cells from all 3 countries. You can see here the cells from Japan, cells from Singapore, cells from South Korea. Thankfully, they all distributed roughly evenly over this transcriptome space. Japan has the largest number of cells in this pilot data set. Singapore has the smallest number of data cells. But of course, this is very early, and now we have actually generated data from, I think, over 200 samples, or over 300 samples, I don't remember. But this is the first handful of samples from each country. And we were pleased to see that we're not seeing any major batch effects. And what do I mean by that? If the blue cells were all on one side, the red cells were all on the opposite side, and the green cells were all in between, for example, then we would say, oops, that's a big batch effect, right? Because it cannot be that the transcriptomes of human beings in Singapore, Japan, and Korea could be that dramatically different. We expect to see some differences, but not that dramatic. But thankfully, we don't see such dramatic differences, so we don't have gross batch effects between the different countries. And then we want to do some clever tricks to reduce even what little batch effect we have in this data set. So that was just a teaser on IDA. There's a lot to do. We have to finish the data generation, generate another few hundred samples despite the pandemic, harmonize the data analysis as much as possible, and then test methods for batch correction, which I'll talk about again a little in a minute. And Dana Pair talked about batch correction yesterday as well. As you can imagine, for any large scale single cell project, you just run smack dab into this problem, right? How do we address batch effects? You simply cannot avoid this problem. One unique aspect of IDA is that we'll be looking at enhancer RNAs, and this is driven by Jay Shin's team in Riken. We're doing 5' single cell RNA-seq, and we expect to discover not just transcription start sites in our data for regular genes, we also expect to discover enhancer RNAs with the same protocol. And then we look at differences within across populations, look at expression Qtls, chromatin openness Qtls, and then see if those Qtls have anything to do with specific diseases. So that's a plan for IDA. And then for the rest of my talk, I'm going to switch over to computational challenges, specifically the challenges of clustering single cells, okay? And I'll illustrate to you how serious these challenges are with this IDA transcriptome data analysis pipeline. Okay. So if you see here, you have a very, very long and complex pipeline for IDA data analysis, starting with the raw data, FASTQ files. We run Cell Ranger, get the gene cell matrix, and then we do some loose QC. We say, you know, if any cell has less than 300 detected genes, we throw it away. And that's essentially a way of discarding empty droplets. If any cell is, you know, half the reads are mitochondrial, throw it away and so on. So that's a very, it doesn't throw away all the crummy cells, but it gets rid of the very worst of it. And then we have a filtered gene cell matrix. And then we run, you know, demuxlet to demultiplex the samples. Because the way we generate the data is we pool cells from multiple individuals and run it simultaneously on the 10x machine. And then we bioinformatically separate those cells out into the specific donors that they came from. And that is Jimmy Yee's demuxlet algorithm, and that's demultiplexing. And we also run doublet finder to remove doublets. And then we have a cleaned up data set, okay? And I would say, so this was our half QC, and then we did a half cleanup, okay, of removing doublets and matching cells to donors. But we still haven't finished the QC. We still haven't finished the cleanup. There's still a ways to go. So then we normalize the data using any of your favorite normalization methods. And then we still have substantial technical variation. So then we cluster cells using our own supervised clustering method, which is quite robust to technical variation, as I'll show you in a few minutes. And based on this RCA clustering algorithm, we identify the major cell types. By major cell types, I mean, is it a T cell? Is it a B cell? Is it a conventional monocyte? Is it a dendritic cell? Is it a non-canonical monocyte. So those are major cell types, right? NK cell and so on. And then we realize, or what we realized a while back, and we recognize that each major cell type will have its own QC distribution. So in the bottom right, you see here, the x-axis is the number of detected genes, and the y-axis is the percent mitochondrial, and each dot is a cell. And you'll see here that the cells, so these black dots, are distributed in your QC space, but there's one central region where they're concentrated. And so we draw a red box around the central region in QC space and say, those are our good cells. Okay, we throw away all the cells that are outside this red box. And we draw this red box independently for each cell type. So we'll have one QC distribution and one set of QC cutoffs for T cells, one set of QC cutoffs for B cells, a different set of QC cutoffs for myeloid cells, and so on. And I'll give you some examples of why that's really, really important. You cannot use one set of QC cutoffs for all cell types because different cell types have different distributions in QC space. Okay? You might be being too strict for one cell type and too lenient for a different cell type. Then having done this cell type specific QC, which is stricter, then we have our final set of cells, which we again cluster using supervised clustering, RCA, to re-identify the major cell types in this cleaned up data set. And then we use Dubstepper, it's a feature selection method, for each major cell type, we do de novo clustering on each major cell type to identify the subtypes. Then you take their marker genes. We use feature selection to identify marker genes and then identify subtypes de novo within each major cell type. Okay. And in this step, we could use explicit batch correction, or we could do it de novo. So even I'm out of breath explaining this whole long pipeline, right? But you just need to take these pains when you're analyzing a large data set and you want high quality results and you want to minimize technical variation in batch effects. Now, a lot of you might be familiar with CCA and MNN. These are explicit batch correction methods. Notice how they come at the very end of this very long pipeline, right? And why do we do that? Because we don't want to be in a garbage in, garbage out scenario. You have to give any batch correction method, you have to give it data that's as clean as possible and as well subtyped as possible so that you're only asking it to take you the last mile. You have carried the ball a long way on your own before you pass the ball to CCA and MNN, right? And sometimes by the time you get there, you don't even need CCA and MNN. So I hope I've impressed upon you that clustering is challenging and clustering is not an algorithm. Clustering is a pipeline. Okay, so if there's one take-home message you want to take, clustering is a pipeline. It's not an algorithm. So where does that pipeline start? It starts with QC. Okay, I want one slight detour also. A lot of people think that clustering is a solved problem. Okay, and that I equate to a famous statement by a former American president, which is a bit premature, saying mission accomplished. Okay, but for clustering, the mission is not accomplished. So saying mission is accomplished for clustering is like saying, okay, I can cluster PBMCs, and when I try to cluster tumor samples, I get some results. The results make sense. Mission accomplished, right? That's not quite true. That's like saying, well, I have a drug that works on cancer cell lines. When I apply it to patients, sometimes the tumor shrinks. Mission accomplished. Cancer is cured. That doesn't make sense, right? So that's setting a very low bar. In reality, cancer is still with us. And in reality, we need to set a higher bar for clustering also, because clustering is not a solved problem, right? When you do cohort scale single cell analysis of human samples, or even mouse samples cohort scale, you have profound data quality artifacts, batch effects, major variation depending on what feature selection you do. Doublet removal is non-trivial. Identifying rare cell types is non-trivial. Defining cell states, which are more subtle than major cell types, very, very non-trivial. And then all of this changes can change dramatically when you change your parameter settings, right? So it's not a push button operation. So let's start at the very beginning of this long pipeline. and that's QC. Okay. So QC is important. What is QC? QC is in this context is just a set of filters. You filter and you refilter and you refilter to your data till only the juice comes out and the dirt is left behind. Okay. And essentially filtering is a process of getting rid of dangerous things that you don't want. For example, you see here, it's very relevant. People use filters on their face to get rid of pathogens that they don't want, right? They don't want the pathogens coming into their airways. And that's very, very important. If you use filters, if you don't use a filter, or if you use filters the wrong way, those pathogens will get in and those impurities will cause problems for you, right? And so filtering is very important, single cell analysis. Here's an illustration, and I should thank Jonito Ignatius from my team who did the data analysis for this, and also my collaborator Ian Tan, and a very large team actually for this project, which is a colorectal cancer single cell project. So here I'm showing you 65,000 epithelial cells from colon tumors and matched normal in QC space. So in QC space, the x-axis you see here is percent mitochondrial reads. Each dot is a cell. Mitochondrial reads percentage in the x-axis, and the y-axis number of detected genes. Now you see here that I'm using conventional QC. Okay, if more than 300 genes are detected, I call it a good quality cell. And if less than say 25% or 30% mitochondrial reads are detected, I call it a good quality cell. Okay. So for conventional QC, everything in here is a good quality cell. But you can see here, there's a dramatic variation in quality in the things that we're calling good quality, right? And in particular, okay, if you zoom into this box over here, which is your conventional QC, you see here that there's a very large population of crummy cells in the lower bottom left corner, which barely have 300 or 400 or 500 detected genes. And then you have an equally large population up here, which have 5,000 detected genes. Do we really believe that these are all of these dots in this box are healthy cancer epithelial cells? We suspended disbelief initially and we explored that, and then we discovered that no, the cells in this bottom left-hand corner are just debris. It is debris from the tumor dying on the pathologist's table with ischemia and then dying during dissociation. So you'll expect a lot of epithelial cell debris and half-dead cells, and that's precisely what you see here in the bottom left corner. And they're heavily mixed with soup. They don't look like proper epithelial cells at all. These cells up above, on the other hand, they look like nice, healthy epithelial cells. Okay, so we do epithelial cell specific QC for our cancer projects, and we try to isolate this population over here in this oval. And those are our healthy cells. We don't do conventional QC. We do QC that's much stricter than normal. And of course, the postdoc, any postdoc would be horrified if you said, I only want these cells. I'm going to throw away these. They'll say, are you crazy? You're throwing away half my hard-earned cells that my blood, sweat, and tears went into this. You're throwing away half my cells. You're crazy. But then analyzing it again and again, I think we all came to the realization, yes, we should throw away half our epithelial cells because these are just debris. Now, here's an example of the benefit we get from throwing away half those cells, okay? Here we're clustering those 65,000 epithelial cells with this very loose QC, okay? So we're clustering all of these conventional loose QC cells. And you can see here the cells are colored by patient. And you can see here that the cells somewhat cluster by patient, but not very well. For colorectal cancer, you expect each patient's cells to form a distinct cluster, but the colors don't really separate out that well, okay? They're quite intermixed, particularly over here. And that is primarily because your entire clustering pipeline is facing garbage in, garbage out. This is the garbage that you're pushing in. This is the garbage that you're getting out, okay? Because these low quality cells, they affect your feature selection. Whether you use highly variable gene feature selection or use some other feature selection method, your algorithm will be biased towards selecting features that separate the debris from the good cells. That's not what you want. You want to separate cells by biology, not by technical bias, right? So I've claimed that this is garbage out, that this is not a good result. So let me show you the good result. Here's a good result. We do stricter QC. We take only these cells. Okay, we throw, And all these cells here we've thrown away. We've thrown away half the epithelial cells. And this is our new cell distribution in QC space. Now you see the cells separate out much more cleanly by patient. You still have a jumble over here. These are the normal epithelial cells. Okay, these are not the cancerous cells. So you expect them to intermingle by patient. But the cancer cells, you expect them to separate out cleanly by patient. And if I just toggle back and forth, you can see here that when you have crummy quality data, the cancer cells don't separate out by patient very well. When you have good quality data, the cancer cells separate out much more cleanly by patient, right? So any kind of biological signal you want to get from your data just gets much sharper and cleaner when you do your QC in a sensible way, in a strict way, I would say heartlessly, right? By throwing away a lot of cells and by doing cell type specific QC. For epithelial cells, we're asking them to have more than 2000 detected genes. But if we do that, we'll throw away all our immune cells in the tumor, right? So our QC box for immune cells is much lower. We don't ask for more than 2000 detected genes in immune cells. And that's why it's very, very important to do cell type specific QC in your primary tissue data. Each cell type will have its own QC parameters. Now I'll show you another example of the pitfalls of conventional QC, okay? So here we're looking at PBMCs, and you can see here that these T cells, these pink T cells, they're all over the place, right? So these are monocytes. These are, let's see, either plasma, I think these are platelets. And there are these T cells bridging monocytes and platelets, which doesn't make any sense. The T cells should all be sitting over here with the NK cells. What's going on? Look at your QC space. Again, you have two populations, right? Two major populations. One with a low number of detected genes, one with a high number of detected genes. We hypothesized that this was debris, and indeed it turns out to be the case. So, debris and doublets of debris, okay? So it turns out that these were all called as doublets by Freemuxlet, and indeed they were low quality cells. So you could kick them out because they have low detected genes, or if you're more fancy, you can kick them out using Freemuxlet as doublets. But the point is that this is low quality junk that you don't want in your data set. And once you remove it, okay, so these are all the cells that you remove, you get much cleaner clustering. Compare the clustering up here, where the T cells are in entirely the wrong place. With your really clean clustering here, now the T cells are nicely localized over here, along with the NK cells. B cells are very far away, platelets are very far away, monocytes are very far away, right? And this again, work of Deepa Rajagopalan and Quaylin in my team in Singapore. So here again is an example of how we do cell type specific QC. So I hope I've convinced you that each cell type requires its own QC, and that you really need strict QC to get sensible clustering. And this is one of those, dirty secrets you can say of single cell analysis that no one really pays attention to. And here's an example again of how the QC parameter distributions, and the x-axis all the cell types, QC parameter distributions for different cell types is very different in colorectal cancer. So please use variable cutoffs in your QC, right? Different cutoffs for different cell types. And then once you've done your QC, then you get into the clustering algorithm. And again, the main message I'll try to give you here is that traditional unsupervised clustering is not very robust, particularly when you have a lot of technical variation. Okay, so I've tried to dramatize that by saying clustering algorithms need adult supervision. What does that mean? So unsupervised clustering, most people don't use the word unsupervised. They just say clustering because they assume that clustering is by default unsupervised machine learning, right? You just look at the data set, you detect some patterns. You don't have any external reference. You don't have any external guides for how to cluster these cells. And so these are some popular unsupervised clustering methods that are applied to single cell data. And this is the work of Florian Schmidt in my group. And this is an example of the pitfall you can have with unsupervised clustering. And Danaper referred to this yesterday. So the red cells come from one protocol, blue cells come from another protocol. So the red versus blue is the batch effect. And you can see here that these PBMCs cluster majorly by batch effect, much more than they cluster by biology, right? So if I remember correctly, these are T cells, these are T cells, and these are myeloid cells, these are myeloid cells. They should sit on top of each other, but they're far apart, right? Or perhaps these are myeloid cells, I'm not sure. So, but majorly they cluster by batch effects. So what does supervised clustering mean? Okay, so this is our algorithm, which Florian Schmidt has updated. So this is RCA2, version 2 algorithm for supervised clustering. We start out with, over here, some reference cell types, okay? So each column here is a transcriptome of a reference cell type in this heat map, and each row is a marker gene of those reference cell types. And we, and then that's one input. So this is the supervised part, okay? RCA uses this reference panel for supervising the clustering. And this is your input data, your conventional gene cell matrix, and each dot is a cell, and each color is a cell type, and you want your cell types to cluster nicely in your gene expression space, okay? But the cells, as I've shown you, they don't always cluster so nicely in your original gene expression space. So what do? We say, wouldn't it be wonderful if we could put these cells into some biological space, okay? What do I mean by a biological space? Let's say in that biological space, the x-axis is how similar my cell type is to cell type, my cell is to cell type 1. The y-axis is how similar my cell is to cell type 2. My z-axis is how similar my cell is to cell type 3, okay? Then you can see here the cells of different cell types separate out very nicely, right? Because the green cells are of cell type 3, they'll all sit over here. Blue cells are cell type 1, they'll all sit here. Yellow cells are cell type 2, they'll all sit here. So as long as you have the right reference transcriptome, or as I'll show you, approximately the right reference transcriptome, you can cluster cells in a biological space that separates them much better, right? And that's what RCA does. It projects these cells into this space of reference transcriptomes and then clusters in this reference transcriptome space. And that is what we mean by supervised clustering. So here's an example of the beautiful results you get from supervised clustering. And this is site-seq data, so you know the ground truth, okay? You can see here that each column is a cell, each row is a reference transcriptome, is an RCA reference transcriptome in the RCA reference panel. And the color indicates how similar the cell is to that reference transcriptome. You can see here that in this reference transcriptome space, you see beautiful, very crisp rectangles of similarity, right? And so the clustering is almost unambiguous. You can identify NC monocytes here, dendritic cells here, naive B cells, plasmacytoid dendritic cells, mature NK cells, effector T cells, and so on. So we were encouraged by this a while back to generalize this method and use it on a number of samples. Now here's the same example I showed you up above, unsupervised clustering, giving you profound batch effects. And this is supervised RCA2 clustering. You can see the red cells are exactly on top of the blue cells. So these are the T cells. These are the myeloid cells. And then you're no longer clustering by batch. Note that we did not do explicit batch correction. RCA did not know that the red cells were from the red batch and the blue cells were from the blue batch, okay? RCA was just given all of the cells as one jumble, and it clustered them without any observable batch effects, or virtually no observable batch effects, because the biological projection space is orthogonal to your batch effect space, right? And this is an example of just how robust reference-based clustering is to batch effects. So that's why we use RCA always for our first pass clustering to identify major cell types like T cells, myeloid cells, B cells, and so on, in the presence of profound batch effects and technical variation. And after that, within each major cell type, within T cells, once you've identified them, you can identify any downstream algorithm you like for subclustering them, feature selection, whatever. So how well does RCA perform compared to other methods? And we wanted to look at two metrics, right? So the x-axis is a silhouette index-based metric of how good the algorithm is doing, how well the algorithm is doing at separating out different cell types. Okay, so you want to be on the far right-hand side of the x-axis. Y-axis is how well is the algorithm doing at rejecting batch effects. Okay, so if you're up here on the y-axis, you have high batch effects. If you're down here, you have low batch effects. So you want to be in the bottom right-hand corner. High biological separation, low batch effect, right? And you can see here for this FACSeq panel of synovial single cells, RCA gives you the best trade-off, also the best biological separation and the lowest batch effects compared to all these more widely used clustering methods. And note that we even comparing to MNN and SC transform, integrate data, Surat integrate data, and so on. So these are explicit batch correction methods. They know which batch each cell came from. They still don't do as good a job as rejecting batch effects and prioritizing biology as this method, which does not know, which uses supervised clustering, but does not know which batch each cell came from. Here's another benchmark where we use site-seq antibody tags and independent ground truth to say what is the biology and what is the batch. And again, you see here that RCA2 sits in the bottom right-hand corner compared to all the other algorithms, regardless of whether they use explicit batch correction or not. Now, in this case, you can see here this green method and MNN correct, Surat integrated, they are all using explicit batch correction. So yeah, they do very well at removing batch effects. In fact, slightly better than RCA, okay, marginally better than RCA. But they do substantially worse than RCA at separating cells out by biology. So that's a problem. So a lot of conventional batch effect removal methods, they overcorrect. They remove batch effects, but they also remove much of the biology, right? And that's because they do not have any reference data set that they can use to say what is biology, what is batch. And that's the advantage of supervised clustering. So supervised clustering in the new RCA implementation is very scalable. You can see here that RCA goes up to hundreds of thousands of cells. And yeah, so this lower curve is RCA2. This upper curve is the old version of RCA, which we're much faster than, more scalable than now. So we benchmarked up to 200,000 cells, but we actually run RCA2 on much larger data sets as well, because it's just uses graph-based clustering downstream of the supervised projection, and graph-based clustering, as you know, is highly scalable. So what's new in RCA2? RCA2 is feature rich. I won't go through all these new features compared to RCA, but suffice it to say it's more scalable, more robust, uses graph-based clustering, and has a number of bells and whistles and useful features that people want to see in a feature select in a clustering algorithm. Now, one question always comes up: this supervised clustering method, what if I throw some data set at it, and in that my data set has cells that are not in that reference panel? Can you really separate them? Okay. Now here's an example. I'll show you that you can really separate them as long as your reference panel is rich enough. Okay. So here on the right-hand side, we're clustering single-cell RNA-seq data from this paper down below from acute myeloid leukemia versus controlled bone marrow. So red cells are controlled bone marrow. Orange cells are cancer bone marrow. You can see, and we did not have any bone marrow cells in the RCA reference panel. We did not have any cancer cells in our RCA reference panel. And yet you see a rich diversity of normal bone marrow cell types. You see all the normal bone marrow cell types. And the cancer bone marrow cells separate out compared to healthy control. So you can see here that not only are you finding healthy bone marrow cell state variation, which is not in our reference panel, you're already seeing that the disease state is different from the healthy state. And that's because your PBMCs actually, and healthy PBMCs have enough variation, enough biological variation in them that even if you project these bone marrow cells into that PBMC space, you can separate out the major cell types and cluster them, and you can identify disease versus control differences. And here's an example of COVID versus controlled PBMCs. Same take-home message. There were no COVID PBMCs in our reference panel, but still we can identify difference in state. All right, I think I've displeased one audience member. So I'll speed up. So just to summarize, the RCA reference panel does not need to have exactly the same cell types as in your favorite data set. It just needs to have vaguely similar cell types. Now, here's an example of where the rubber meets the road. Okay, what can you learn with supervised clustering that you cannot learn with conventional clustering? Some of you might be familiar with this data set from Whale et al. It's a really nice data set, MCs from COVID patients. And they identify this peculiar developing neutrophil population that seems to be emerging from plasmablasts. Okay, that's odd because plasmablasts are lymphoid cells and neutrophils are myeloid cells. So this violates your normal hematopoiesis developmental ontology. Okay, so myeloid cells should not be emerging from lymphoid cells. But they hypothesize that in COVID, perhaps lymphoid cells can transdifferentiate into myeloid cells, forming these developing neutrophils. Okay. We hypothesize something different. We hypothesize that this was an artifact of very generous QC, and I've talked about that before. So if you do very generous QC, you have a lot of technical variation. Then you do unsupervised clustering, as they did here. Then unsupervised clustering is very vulnerable to technical variation, and then you get weird artifacts in your clustering. And we hypothesize that this was a weird artifact of clustering with some kind of confluence of data quality variation and unsupervised clustering. So we said, okay, let's do supervised clustering. Let's take exactly the same data set. Don't change the QC, but do supervised clustering. Okay. And now we see here that I'll show you where the developing neutrophils are. Okay, so the red cells are the cells annotated as developing neutrophils in that paper. A lot of these red cells in our clustering, in our RCA2 supervised clustering, we felt they were simply conventional monocytes. Okay, and then some of them were genuinely a distinct population, and some of them were actually here. These gray cells, actually low quality cells and doublets. Okay. So notice two things. These developing neutrophils are no longer connected to plasmablasts. So plasmablasts are over here. There's no longer a bridge connecting plasmablasts to developing neutrophils. If anything, there's a bridge connecting developing neutrophils to myeloid cells, which makes sense because developing neutrophils are myeloid cells. And that bridge is actually low quality cells, okay, which we want to throw out. But still, despite the presence of these low quality cells, supervised clustering gives you a sensible clustering output, a sensible UMAP and so on, where there's no transdifferentiation of plasmablasts to myeloid cells, okay? Now, what happens if you throw away these low quality cells? Perhaps now, with less technical variation, the clustering problem becomes easier and your unsupervised clustering method will be able to correctly cluster these developing neutrophils. Indeed, that's what we found. Now, this is stricter QC and unsupervised clustering. Again, you see here that your developing neutrophils, they form a distinct cluster. They are somewhere near monocytes. They're very far from plasmablasts. Okay, there's no bridge connecting plasmablasts to developing neutrophils. That bridge was composed entirely of low quality cells that were distorting the unsupervised clustering. So now you see a more conventional picture. So you can either do strict QC and unsupervised clustering, you'll get something sensible. Or you can do generous QC, tolerate a lot of low quality data, but do supervised clustering. Then again, you'll get a sensible picture. But don't do generous QC and unsupervised clustering, particularly if you have a lot of technical variation in your data. So RCA2, supervised clustering is our default clustering algorithm now. Before cell type specific QC, and as the human cell atlas defines more reference transcriptomes, RCA2 will become more and more powerful, identify more and more cell types. And now we're testing it on over 1 million cells. And in principle, there are no limits to how much it can scale because it can scale as much as graph-based clustering. Okay. The last thing is once you've identified your major cell types, this is a major cell type, there's a major cell type, there's a major cell type, there's a major cell type, and so on, using supervised clustering, you want to go inside and identify subtypes, CD8 TM, CD4 TM, CD4 naive, and so on. So that's where you do your unsupervised clustering. And to do unsupervised clustering, you need feature selection. You say, which genes am I going to use to cluster these, to subcluster these T cells? Now, conventionally, people use what's known as the highly variable genes method. Virtually everyone in the field uses variations of that highly variable genes method. And we said, why not use something different for feature selection, okay? We use the property, now I'm just showing you an illustration again from PBMCs, and this is work from Bobby Ranjan and my team. This is the gene-gene correlation matrix, okay? So marker genes in PBMCs, you can see here that they correlate with each other. These red boxes indicate groups of marker genes that are correlated with each other. And so that is a basic property of marker genes. On the right-hand side, you'll see non-marker genes. They correlate with themselves, but they don't correlate with each other across cells, right? When you look at the gene-gene correlation matrix. So we said, let's use this property of marker genes for feature selection. Let's use gene-gene correlations, okay? I don't have time to go through the details, but Dubstepper basically does stepwise regression on the gene-gene correlation matrix to identify your initial set. These red genes are your initial set of seed feature genes that you use for clustering. And then those initial set are these red dots over here in your gene-gene correlation space. Then you start adding genes that are neighbors of your initial set based on gene-gene correlation. Then you get your final set of genes, and you use that for clustering. Instead of highly variable genes, you use these red dots from gene-gene correlations for clustering. And we benchmarked it on multiple data sets. So this is the summary on the right-hand side of your clustering quality, okay, as measured using the silhouette index. How good is your clustering as a function of number of genes selected? For when you average across a number of benchmark data sets, you can see here that Dubstepper, that's a red curve, does better on average than the other clustering methods, other feature selection methods. And we compared to a whole range of other feature selection methods. So correlation-based feature selection is quite robust, regardless of how many feature genes you choose. Plus, interestingly, you can see it peaks at around 200 or 300 or 400 genes, right? So you actually need only a few hundred genes to cluster your cells well. Conventional feature selection methods pick 2,000 or 3,000 genes. You don't need that many. You can do better with a smaller number of genes. And Dubstepper also identifies rare cell types quite nicely. Now, the even more interesting thing for us was that Dubstepper was able to cluster single-cell attack-seq data. Now, why are single-cell attack-seq data really difficult for feature selection algorithms? So in fact, conventional single-cell attack-seq data analysis pipelines do not do highly variable gene feature selection. In fact, most of them don't do anything that we would recognize as feature selection, right? And that's because the number of reads, attack-seq reads in each attack-seq peak is usually either 0 or 1. It's digital, okay? At most, it'll be 2, but usually 0 or 1. Now, for data that are 0 or 1, right? Imagine a gene expression matrix in which all of the entries are either 0 or 1. Your highly variable gene feature selection method simply will not work because the mean will exactly predict the variance for each gene, right? So what can you do? Well, correlation-based feature selection can still work just as well, right? Even for a matrix of 1s and 0s, gene A can be correlated with gene B, or gene A can be uncorrelated with gene B. So we just naively applied Dubstepper as is to single-cell attack-seq data. And here you can see that it gave you very nice clustering of this bone marrow data set where hematopoietic stem cells nicely differentiated into an erythroid lineage, a granulocyte monocyte lineage, lymphoid lineage. If you don't do feature selection, you get a slightly chaotic picture. You don't recapitulate that differentiation hierarchy. Or if you just brute force apply highly variable gene feature selection, again, you don't get clean separation of your developmental lineages. Right? So we're very excited about using Dubstepper for feature selection, even in single-cell attack-seq data. So to summarize, correlation-based feature selection is a viable alternative to highly variable genes. Dubstepper is already available on GitHub. And by the way, RCA is also available on GitHub, freely downloadable. And the preprints are all available on bioRxiv. You can cluster with relatively few genes. Dubstepper is also scalable to over 1 million cells, and it's also applicable to single-cell attack-seq data. So hope I've given you a flavor of some of the challenges of single cell clustering and how it's a long road to get good results at the end of it, but it's worth it, right? And I've mentioned all my collaborators, or most of my collaborators here, the people doing the work. Thank you. Happy to take questions.
Thank you so much, Shyam. That was a perfect talk for the purpose of this meeting. I hope everybody enjoyed it as much as I did. We do have quite a few questions for you. So I'm gonna start with the ones that were voted the most from the audience, okay? So the first one is from Tiago Lubiana Alves. A question on filters. I always wonder which values to use, for example, the mitochondrial percentage cutoff. As you mentioned, each cell type favors different QC values. Do you have guidelines or tips for choosing good cutoffs? Do you redo QC after first round of clustering?
Exactly. Thanks for that question. So here's our pipeline again. We do a first round of, we do lose QC over here, which is, the same cutoff for everyone, just very generous cutoff. Then we do the first round of clustering using RCA because supervised clustering is quite robust to data quality variation. Then we identify the major cell types. Then we visualize the QC distributions of each major cell type individually. And then we manually pick these red boxes. We could come up with an AI algorithm to pick this red box of our second QC. I don't think it'll make a huge difference. And then after our second QC, we start clustering all over again. So just like you said, we do our first clustering, then do cell type specific QC, then we do clustering all over again on the cleaned up data set. So we use visual inspection. That's a short answer.
Great. They're asking here if the full clustering pipeline is available to be reused or cited, or how does that work?
Excellent question. I think we should make it available. It's probably not user-friendly enough for anyone to use yet. So the individual pieces, RCA2 is very user-friendly. GitHub has got tutorial and readmes and all that. Dubstepper is very user-friendly, got tutorial, readme, etc., etc. But this part is kind of a manual pipeline, and we do want to maybe containerize it and make it usable. So thanks for that question. Unfortunately, we're not there yet.
Thank you, Shyam. From Tiago Lubiana Alves, there is another question here on the Human Cell Atlas specifically. So super interesting topic. He says, thank you. Here in Latin America, people from native ethnicities are often underrepresented in the research community. In HCA Asia, do you see similar situations with any population? If so, are there plans to include people in the analysis or interpretation of results beyond just sample collection?
Wonderful question. And this really gets to the heart of the equity issue. So it's not just about sample collection from people of different ethnicities, right? So that's the representation part of equity, meaning we want all populations to be represented in the HCA, but we also want all populations to participate to the extent they can in the research. And that's the participation equity. And so there, a principle of IDA is we make very strong efforts to make sure that people from each study site generate their own data. We're centralizing the data analysis to some extent, only the primary data analysis. Okay, secondary data analysis is again federalized, but we strive for participation equity. So each team does its own data generation and does its own downstream data analysis. And then there's of course a benefit equity, and that is how does that community that you're representing, how does that community benefit from the research? And that is a question that we cannot answer for them. It's a conversation with each community saying, what are your goals? How does this benefit you? Will your national funding agencies fund this research, and why? And then we try to design it so that it aligns with the goals of each team. And we have, actually participation in IDA has helped at least one team from one country get national funding, because they leveraged their IDA funding to get matching funding from their national funding body. So we do try to ensure equity of all forms. Malays, for example, are underrepresented in international genetic databases. Thankfully, Singapore has a sizable Malay population. Indians are not as underrepresented, but they are underrepresented. Chinese ethnicity are more well represented, but still underrepresented. Of course, Japanese, Koreans are also underrepresented. So those are the initial 5 nationalities. We are expanding Russia, Saudi Arabia, and hope to add many more countries as well to IDA.
There's a related question to this one here from Raul Arias Carrasco. How can one estimate an ideal number of samples or subjects per tissue to elaborate a representative HCA based on the diversity of the population? Or do we have any idea about that so far?
That's a great question. And to some extent, we have to answer that question empirically, right? As in, well, we have generated data from so many cells, from so many people, and we've seen all of these interesting things. Maybe we'll see more interesting things if we sequence more. We don't know, right? So all we can say is what's not good enough, right? So say 50,000 cells from 3 countries is nowhere near good enough, even for something as relatively simple as PBMCs, okay? So let's take the example of Qtls. If you want to do single cell Qtl analysis, even if you do 100 samples from one population, it's not enough. You can do 1,000 cells from one population. Sorry, 100 samples from one population. You can do 1,000 cells from one population, as Joseph Powell and Jimmy Yee have done, and you'll still identify only a few hundred eQTLs from each cell type, right? So you really, I would say you need to hit around 1,000 to get the genetic diversity and genetic associations from your single cell data. And 1,000 samples from one population is a lot. So there's a long way to go.
That was a great answer. Now we have a bit more, some technical questions. Luis Geraldo Fernandez asking, nice talk. How do you define a good threshold for not G?
Sure. So I hope you've seen from those scatter plots that the thresholds kind of self-emerge. You can fit a Gaussian mixture model to the distribution of your cells in QC space. You can do clustering, density-based clustering of the distributions of your cells in QC space. And then each cluster will be a potential good quality cell. So your cluster in your top right-hand corner, top left-hand corner will be your good quality cells. And your other clusters that are lower down with lower NODG are iffy quality cells. They may be cells of a different type, or they may be debris. You just have to examine them one by one and say, each cluster in QC space, does it look, if I look at its markers, does it look like a real cell type? Does it look like soup? Does it look like debris? Does it look like doublets? So you can use a Gaussian mixture model. You can use a density-based clustering model in QC space. We just do eyeball, like the human eye is very good at solving clustering problems in 2 dimensions. And our QC is in a 2-dimensional space, right? Very hard to beat the human eye at clustering in 2 dimensions.
All right, that's from Devika Agarwal. Have you compared RCA2 with Harmony? In recent studies, integration of data sets and removal of batch correction has been widely preferred.
Not yet. I'll defer to Dana Pear, who showed results from Multilucan's bioarchive preprint, where I think she was proposing that Harmony overcorrects batch effects. So, it looks very good, meaning the batches are right on top of each other. But actually the downside is that the biology is also overcorrected, right? I don't take responsibility for that conclusion. That's Malta's conclusion and Dana's conclusion. So you can take it, from them rather than from me. But we would like to add harmony as well to our batch correction methods.
From Tanya Sepulveda, in case you decide to do a strict QC, would it be better to do a supervised clustering? Or would you have the same result as doing an unsupervised clustering?
So the challenge is initially, if you have tumor data, you cannot do strict QC right at the start. Because if you do strict QC, you'll keep the epithelial cells and fibroblasts, you'll throw away the immune cells, right? Or if you do strict QC in the other direction, you'll keep the immune cells, you'll throw away the fibroblasts and epithelial cells. So initially you have to do loose QC. And because you've done loose QC, you have to do supervised clustering, right? To get robust results. Once you've done the supervised clustering, then you can do loose, stricter cell type specific QC. And then for each cell type, you're free to use unsupervised clustering.
Another one on this matter, in which cases should we use integration of the data, and how is it that helping to improve the clustering process?
So we use data, integration data, and MNN and so on, or Surat integrate data. We use it after we've done all our QC and cleaning, after we'd identified major cell types using RCA. Then we take each major cell type and then say, okay, for that major cell type, I have 20 batches. Do I need batch correction? Okay, first I just do unsupervised clustering using dubstepper and some graph-based clustering. If I'm seeing major batch effects, then I'll say, okay, I have to explicitly correct it. But hopefully by then there won't be batch effects. If you have batch effects still, then you say, I'll do a T cell specific harmony, for example, or T cell specific MNN or CCA. And I'll do a B cell specific harmony, MNN, CCA, and cluster them separately. We've done that as well. But it's really trial and error. So sometimes these batch correction methods will completely kill the biology and you'll get results that don't help you. Sometimes they'll help. There's no magic answer. There's no one size fits all.
Okay, we're running out of time. Just gonna drop in another question from Eduardo Duran on the HCA specifically. He's from Chile. He's asking, what would you consider are the major challenges to create the HCA with such diversity from Asian countries? And specifically also if China is part of the HCA Asia.
Let me take the second question first. So China is part of HCA Asia, but China is not part of the CZI grant proposal that we submitted for IDA. So IDA is not HCA Asia. IDA is one flagship program within HCA Asia. And China is a key part of HCA Asia. China hosted the last Human Cell HCA Asia meeting, actually, and also hosted by Goji Go and colleagues. And then the other question is, what are the challenges in doing HCA in Asia? A good example, legal restrictions. So it's hard for China to host their data on a foreign server. And then in various other Asian countries also, you may be allowed to host healthy sample data on foreign servers, but not disease data, or maybe healthy sample data processed, but not the raw data and so on. So certainly the fact that each country has its own ethics and legal regulations makes it challenging. Then simply, whether it's Asia or Latin America or anywhere else in the world, getting teams in 5 countries to use the same reagents, the same blood collection tube, the same protocol for collecting blood, isolating PBMCs, storing them, resuspending them, etc., very, very challenging. It's not trivial at all. You have to put a detailed protocol on, Google Drive or wherever, disseminate it, have lectures about it, and then people will still not do exactly the same thing in each country. Then you'll realize that you have to reharmonize. Then, do you sequence the data all in one place, or do you ship the DNA to another place? There's ethics restrictions there as well, legal restrictions there as well. And then downstream, how do you harmonize the data analysis? So a lot of logistical challenges in harmonization that are not unique to Asia, they're universal, but then of course, each country in Asia will have its own unique wrinkle on this because of its own unique laws. Besides that, I would say we have not had any Asia-specific challenges in this project so far. Okay, I'll say one challenge in Asia that's slightly different from, say, the US or Europe. In Asia, the national funding bodies want to fund translational research generally. They fund basic research, but much less than in the West. So how do you do a human cell atlas type project, which is very, very basic research? It's, in itself not translational. How do you get national funding for that? That has been a challenge for us, and it's possible that could be a challenge for Latin America as well. I'm not sure.
Yes, I think we share very similar challenges, that the funding situation is very similar for us too in Latin America, and diversity of regulation systems and following protocols. I'd say even within the same country, it's complicated. So we can, of course, know how complicated it is to have a streamlined way of doing that in all countries. So also regarding access to reagents and the materials, that is also very variable.
And COVID makes it worse, yeah.
Yes, now it's a different story, even worse, yeah, of course. Cheyenne, unfortunately, we have to go because Hanifa is already here, and we are exactly on time. Thank you so much. We know how difficult it is for you to join us from Singapore, right? So it's really great that you were able to join us today. Thank you so much.
My pleasure, Patricia. Thank you.
We're gonna move on. Our next speaker, Dr. Muslifa Hanifa, is already here. I'm going to introduce her. So Professor Hanifa is one of the 3 Human Developmental Biology Network coordinators. She's a dermatologist with a research background in immunology, and she has pioneered the application of single-cell genomics to study human development. The main focus so far of her research has been on decoding the developing human immune system. Thank you so much for joining us today.
Thank you, Patricia, for the introduction, and I hope you can see my full screen. Please let me know if that's the case. Brilliant.
Yes.
So what I think, bless him, Sham, he's kind of like, it's about 12 midnight in Singapore or something like that. And so he started the talk, and really from once you get the data and what do you do with the data in terms of the analysis. So there's a slight back to front element to my talk, which is I'll be focusing on the upstream component, i.e. up to the point where you get the count matrix for downstream analysis. And that's what my talk will be based on. And before I get into the nitty-gritties of this, is just to kind of give a very broad overview and my sort of like simplistic understanding of, the world of single cell omics, and really the kind of like the departure from more traditional methods is to imagine the tissue or whatever organ that you're interested in studying as a fruit salad. And what the single cell methods allow you to do is to deconstruct the fruit salad to its kind of individual components. And in the past, it's not to say that you couldn't do any single cell analysis, because, by imaging, by flow cytometry, we were doing single cell measurements, but it was just that, limited to specific or limited parameters. With imaging, it might have been limited to sort of 4 or 5, but there are more imaging methods now that are increasing the parameters that you can measure. And with flow cytometry, again, the limitation is how many antibodies or how many antigens can you measure with the fluorescent tagged antibodies that you're using. And with respect to transcriptome, we had to resort to sort of isolating cell types or taking the entire tissue as a homogenate and then measuring the RNA of either the kind of like enriched populations or the tissue itself. So it didn't quite give you the sort of single cell resolution that you would get in being able to measure lots of parameters in individual cells and of lots of cells. So there are several kind of components to it. And if you look at publications that have the word single-cell RNA sequencing either in the title, abstract, or text, I mean, that's just gone up, exponentially since the technology was first developed. And the blue line shows you the cumulative publications since measured at 2012, and the numbers are actually the number of manuscripts per year. So this is pretty phenomenal in terms of what the technology and the uptake of the technology is by the research community. The second thing to consider is what types of information you can measure. And you can see here the different technologies that allows you to either measure RNA alone, protein and RNA, DNA and RNA, epigenome and RNA, epigenome and protein. And this might lead you to sort of like ask the question of what is best to measure. One kind of conceptual way that I have of thinking of this problem is to, if you imagine yourself having different types of scan. You can have an MRI scan, a CT scan, a thermal scan, or just a photograph of you. They all give different information, and one is no more truthful than the other. It's just different facets of an individual or a cell. And that's how I see this, in that the DNA really gives you a lot of the information about the history of the cell, protein, the immediate past and present, RNA, the present and future, and epigenome, the future and the potential. So all of these informations are extremely helpful in trying to enable us to understand about the cell in that particular time that we analyze. My earliest adventure into single-cell omics was with this project, which was published back in 2017 in collaboration with several researchers then at the Broad Institute, and some of them still there now. And this was basically taking peripheral blood dendritic cells and monocytes, and then using, in those days, SmartSeq2 to measure the transcriptome of these cells, and to ask the question, can we then identify or label, or can we detect more heterogeneity in these populations? What else can we discover about these cells that a lot of people have studied? A lot of people know the surface markers that the cells express. Is there residual heterogeneity, and what can we learn from those studies? And really, the type of approaches to generate suspension single-cell data sets have changed dramatically. It's first started off with manual or individual cells being picked out for single-cell RNA or PCR, and then you could take FACS, as we did, and sort individual cells into individual wells, and then you can measure the RNA of those cells. And there are many different platforms that enable you to do that. And then these sort of like integrated fluid circuits, like the C1 fluid ion system. And then now more and more with the droplet methods, because these are sort of low throughput to when you look at it in the context now, allowing you to measure initially, tens or hundreds of cells, so thousands of cells, to probably pushing the limits of tens of thousands of cells. But with the advent of droplet methods, whereby you can capture a cell in a droplet, so that you can do the PCR reaction in a droplet, that has really scaled the number of cells that you can analyze in one experimental setting, and the number keeps going up. And there are all these sort of micro-well techniques to capture individual cells in these little wells. And also, where you can barcode and split the number of cells. So really increasing scalability, and they also come with different costs associated with the different platforms. So that really captures the sort of approach where you can measure lots of parameters in single cells, but notably this process involves deconstructing that tissue, so you can't actually see where the cells are in situ, so you don't actually know, it deconstructs it, so you don't actually know which piece of banana or orange is next to which other fruit. So, you know, what you can do is to reconstruct it based on the single cell data by, you know, kind of more complex computational analysis, looking at, you know, taking into account of local tissue microanatomy, whether, you know, interaction is feasible, looking at receptor ligand expression of those cells when you do the data analysis to say, you know, these cells appear to be interacting. Do we know whether they're within the same tissue microanatomical compartments, so on and so forth. But really to better understand microanatomical niche and locate these kind of cellular interactions and cellular conversations, you know, spatial approaches, spatial omics approaches have come into play, and it really complemented the type of things that you can do. This is just, you know, how you can look at the expression of RNA at tissue level. And here you can see these sort of like, you know, spots where you can visualize the gene expression. And when you actually, you know, try to do this, what you find is that, you know, the original, that's, you know, by, for example, spatial transcriptomics methods, the original spots were about 100 microns in diameter, which has about, you know, probably about 20 cells if you think about a cell as being 10 or 20 microns. And then there's a gap between it. And then the more recent versions, the spots have become smaller, and now you're capturing about 5 cells there, and the distance between the spots have also become smaller. So this is where you've got like, you know, you're able to capture the RNA in tissue that is sort of like, you know, placed on the chip, and the kind of like RNA is captured, and then you can sequence. So it gives you whole transcriptome, but it's not quite single cell. And I think more and more these type of spatially resolved transcriptomics, you know, there's a lot of progress being made, and you can see here the different types of technologies which, you know, really are taking the distances further down and therefore enabling, you know, whole transcriptome measurements at single cell resolution and spatially. And that is to also be contrasted. So that's tissue sections. So that has to be contrasted in the type of measurements that you can make of whole organs, big chunks of tissue, or even whole embryos. So this is another method whereby you can use tissue clearing, and then you can, you know, do either protein or RNA sequential measurements, either by antibody or in situ hybridization, and using light sheet microscopy to measure those antigens or RNA of interest. And you can then construct a more kind of a bigger scale, but, you know, fewer parameters at this point with these type of technologies. And this is what I mean by, in terms of the genome scale versus the resolution, i.e. whether it's single cell or otherwise. And, you know, the kind of like, you can measure many more parameters, but not necessarily have the single cell resolution. And by and large, as you approach single cell resolution, you tend to lose the, you tend to limit the number of parameters that you can measure. So, you know, less of a genome scale measurement. And ultimately, the idea will be to kind of like integrate all of the different modalities, either by suspension, spatial, you know, tissue level, organ base, three-dimensional sort of information, and taking the kind of computational technologies and approaches as well to interpret this data and to say, what is this, you know, what are these data sets and information telling us? What are the biological insights, and how can we apply that to understand the human body and also to better understand disease? And of course, it's going to require lots of different expertise, you know, very much a multidisciplinary team effort, you know, bringing in biology, computational, clinical specialties, and technologies. So this is the sort of like approach that, you know, the Human Cell Atlas initiative has taken to better understand the composition and identity of the individual cells and how they may function that makes up our body. So you can see the enormous application of these type of technologies, not just to understand the kind of like a healthy tissues and organs, but disease mechanisms and as part of the discussion earlier on with Sham, human genetic variation, therapeutic screening, organoids, and also cross-species comparison. So let's get to the nitty-gritty of how do we generate the data and what are the kind of some of the considerations that we require. So I'm just going to give an example, and most of my talk is going to be based on how do we generate suspended single-cell RNA sequencing or single-cell omics, multi-omics data sets from dissociating cells from tissue. So here's an example of, you've got a patient, and if we were to take skin, you take a biopsy of that skin, and you've now got this tissue sample. So what do we do in terms of the steps? So firstly, you have to dissociate the cells, and then you have to capture the single cells. And then I'll go through the different steps in more detail with the talk. So here's the tissue, here's the cells. And once you've dissociated it, you can either mechanically dissociate it or you can enzymatically dissociate it. Now, dissociation has to be adapted to the organ that you're interested in. And it's very different to dissociate, for example, the spleen, even if you were to compare the mouse spleen and the human spleen, or dissociating tissue such as skin, which is a lot more collagenous. Liver is another example, which is more similar to spleen, whereby the cells are sort of, almost like a loose aggregate with lots of sinusoids. So you need to balance, a method that's going to give you good yield, i.e. as many cells as you can get dissociated, and also that's going to give you good viability. So in the process of dissociating all the cells rapidly, that you kill the cells, or that basically you just don't capture the cells that you need to capture. And then you can either load the cells onto any of those platforms. So I'm going to mainly focus on the kind of like what most people are using right now, the commercial platform 10x Genomics. Or you can either take those cells that you've dissociated, enrich for live cells or enrich for the specific cell type that you want. And maybe you can do this by max or fax, but bearing in mind as well that when you dissociate the tissue, some of these enzymes can affect the surface expression of those antigens that you're then going to use to enrich for the cell type. So, a lot of kind of careful consideration and expertise in terms of the tissue and how to handle the tissue is required. So this is, as I say, the kind of like platform that I'm going to mainly focus on in terms of, getting the cells and loading them in here so that you can actually make the emulsion with the kind of like droplets before performing the PCR step to make cDNA from the RNA and then to amplify the cDNA. So one other thing that you can also do before you actually load the instrument is to then leverage the ability to measure both protein and the transcriptome, the whole transcriptome of the cell. So one of the applications include an antibody panel stain where the antibodies are tagged with primers so that you can sequence the primer associated with a particular antibody as a surrogate measurement of the surface protein expression. Or you can take the cells or the tissue and snap freeze, which you can then homogenize and dissociate and extract the nuclei before taking it on to the next step. So in the first option where you incorporate an antibody panel staining with antibody conjugated with primers and staining the cells before the 10x platform allows you to kind of measure the protein and transcriptome, and that's called the sort of site-seq type technology. And you can use an optional cell hashing, #oligos, to basically allow you to label individual samples so that you can then pool them together before loading, which allows you to analyze many more cells from lots of different individuals and bringing down the experimental cost because you can use the same amount of reagents to analyze many more cells. Or you can take the step whereby you extract nuclei for nuclei RNA sequencing. So these are the kind of like different options that you have, and I just want to sort of like talk a little bit about, you know, the considerations. And I think, you know, we touched upon that before. You have to think about how many cells that you want to analyze per individual, and, you know, what's the feasibility of can you store the sample before analyzing, or do you need to analyze the cells fresh? You know, how many cells you need per patient to make a meaningful comparison. And that's all going to be dependent on what your research questions are, you know, and what you want to get out of the data. So so the purpose of the kind of like loading the cells here is to sort of make the droplets, you know, to generate these gems. And then you can actually, you know, do reverse transcription, which is essentially, you know, once you've convert the RNA to cDNA, and then you can amplify the cDNA, and you can, you know, clean it up so that it's pure, and then you can look by bioanalyzer whether you've got a good peak, and this is just a representative trace for peripheral blood mononuclear cells. And then you prepare the library, you know, you index samples, pool them, and then you sequence. And then, you know, once you've done the sequencing, essentially the next steps involve running through, in this case, the cell ranger pipeline to align to a reference genome. And then the output are the sort of things that you get, which is the count matrix. And then you go through the steps of quality control, clustering, identifying doublets, and annotating the cells, and so on and so forth. I'm not going to focus so much on that, but just to mention as well that if you pool your samples, either, and you need to subsequently, you know, with the hashtag oligos or with the genotype, you can then demultiplex to work out where, you know, from your combined several donor analysis, which cells are derived from donor A, B, C. So you know, that's something to consider. So I'm now going to show you some of the examples of how we've approached the data generation and the considerations that we made in terms of the data generation. So one of the areas that we've studied is the developing human immune system. And here you've got lots of different organs. And you find that, you know, some, you know, how, you know, if you were to study all of these organs from one embryo, one fetus, can we use the same, you know, enzymatic dissociation protocols? Will we get the same cells from each of those organs? Are there specific cell types that, you know, are lost with one dissociation protocol and otherwise? So we spent a bit of time optimizing this and settled on one that we thought, you know, we could use consistently. And we found for, you know, particularly like the liver, you know, and the spleen, you know, you just really mainly is more mechanical than enzymatic. It's very, very low enzymatic. And for the bone marrow, it doesn't really need any dissociation, but more like crushing the bone marrow. And you know, in contrast to the skin or the kidney, which are slightly different. So there was a bit of optimization and a bit of consideration of how we would approach the different tissues and different organs. And the second question, which I alluded to before, is, you know, are you interested in analyzing all of the cells or specific cell types? How many cells? And that has to be balanced with what are your research questions. If your research question is to, you know, understand how the, you know, entire immune, you know, cells and network are, you know, is assembled together, then you want to analyze as many cells as you want. You know, and this is a good example of, you know, migratory birth patterns that you wouldn't necessarily appreciate if you were to just analyze one lineage or very few cells. It's only when you see this birth pattern when you have lots of births. So it'll be the equivalent of like the number of cells that you need to study to be able to sort of like be able to pick up those properties and answer the questions that you want. So some specific examples. So this is the study of human fetal liver, which we published 2 years ago. And what we wanted to do was to say what happens during fetal liver hematopoiesis. And we looked at samples between 7 to 17 post-conception weeks. We also had other organs, and I talked to you, and I mentioned earlier about how we optimized the tissue dissociation protocols to see what would give us the best yield and viability. And we talked about, you know, how many cell types and how many donors that is required. So we divided the gestational stages into different categories and had about 3 to 4 donors per stage, and that gave us about 19 donors in total, and we analyzed about 140,000 cells. And there are various different ways of doing this. You know, you kind of like find that you're not finding out anything new anymore with increased cell numbers, but also to be able to have the, you know, robust, to be able to analyze the data in a robust statistical way. So these were the sort of like, you know, our early studies. And in fact, I want to bring this here because if you look at, you know, some of the batch correction methods, how do you know what is batch effect versus what is just changes, biological changes with gestation? So there's quite a lot of interpretation and actually understanding the data rather than just sort of blindly following protocols, because, you know, what you don't want to do is throw the baby with the bathwater. By batch correcting, you're removing all of the biological information as well. So this was the strategy we used. So we dissociated the cell, we isolated the cells. And then what we did was we labeled the cells with CD45, and then we used FACS to isolate live cells. And the reason we did this was, at that point, we weren't sure whether during development, all of the hematopoietic cells would be CD45 positive. So we didn't want to throw anything away, because there may be some cells that are during development that haven't acquired the CD45 expression. Secondly, we weren't sure if a particular organ was super enriched or not enriched for immune cells. If we were to just take all of the cells, you know, would we actually, would we be able to detect rare populations? So for example, in the non-hematopoietic fractions, we might have some rare, you know, pericytes and such like. And if we just took all of the cells, it would be proportional representation. And by doing this, but putting the FACS gates contiguously, so you don't throw any cells away, you capture everything, you can then have a whole lane which you can run just CD45 negative, or a whole lane that's CD45 positive. At least that's what we did in this particular experiment. And then for the SMART-seq2, we had an additional marker, MHC class 2 or HLA-DR. And the reason is it would be enable us to take the HLA-DR population, which are CD45, and they will be the antigen presenting cells. And those that are just CD45 positive and HLA-DR negative will be the lymphocytes. So this way, you know, you will capture some of the rare subtypes, and you can do, you know, many variations of this. But, you know, at the end of the day, it's kind of like what research question you're asking. And so you can kind of like do the downstream analysis. And the point of this is not to show what are the different cell types that are there in the fetal liver, but to kind of say to you that, you know, sometimes you don't find a particular cell state. In this case, there were no neutrophils in the fetal liver. And one of the things to consider is whether the platform that you're using can capture all of the cells. In this case, we knew that, you know, the neutrophils weren't there because the fetal liver hematopoiesis does not support neutrophil differentiation. It occurs much later after bone marrow, during bone marrow hematopoiesis. But it's worth knowing what are the limitations in the same case as if you were to analyze human peripheral blood mononuclear cells. Then if you use lymphoprep or FICOL, you won't be capturing polymorphs and, you know, granulocytes, because they would be, you know, they would have been removed in that process. So just knowing what you want. And always, it isn't just, you know, for me, it's not good enough to just be able to say, here are the cells, these are the RNA expression, and these are what they are based on their RNA expression. You know, it's always useful to have orthogonal validation methods that allow you to then say, hey, you know, the findings from the single cell RNA sequencing, you know, we validated it, and we saw that, you know, we could identify the cell states or cell types that we found, you know, using a different way. So in this example, what we did was we took the single cell RNA sequencing data, identified the best RNA markers that, you know, to design a flow cytometry panel, and that allowed us to isolate the cells, prepare cytospins, and then stain them with GIMS, histochemical staining, and then, you know, be able to look at the morphology and morphological assessments of blood and immune cells. is well established. It's used in clinical pathology to identify hematological malignancies. So leverage that as a way of sort of saying that, what we found was indeed, accurate, and we could validate it. So we saw these different stages of erythroblasts in their differentiation, and using this morphological assessment, that was one way of validating our findings. So, look at multiple orthogonal methods. So my next example is going to be focused on human skin. And again, it's not just one platform, lots of different orthogonal approaches to sort of validate your using, the single-cell RNA sequencing approach as a discovery platform. These are your validation platforms. And you can then start making comparisons across several tissues. So looking at healthy and contrasting it to understand disease, and also contrasting it with prenatal first trimester skin to understand what's the difference between human skin during development and in adult life. So I've kind of like highlighted this because I just want to show that, you might think actually, that, in atopic dermatitis, we've got 4, in psoriasis, we've got 3 donors, and in developing skin, we've got 7 donors. So is 4 enough? Is 3 enough for a particular disease type? It all depends on your research question. For us, when we started doing the healthy skin work, we didn't really find that we were detecting a lot of heterogeneity. And what we wanted to do was to compare with, a disease state to see how they, what were the cell states that were present in healthy and disease. But we had a very strict criteria of what type of disease tissue we were going to analyze. It wasn't just anyone with the disease. It was basically the criteria was that the rash would be on the trunk, it would be an acute flare, they wouldn't have had any biologic treatment, they would have stopped any systemic treatment for the last 8 weeks, and they wouldn't have had topical steroids for 1 week. And by doing that, by making your criteria quite strict and narrow, you make your tissue sample quite homogeneous, or you limit the heterogeneity, and that allows you to make comparisons in a more meaningful way. So again, go back to the type of questions that you want. And for the developing skin, this was 7 from first trimester skin. So in the consideration of skin, you have the epidermis and you have the dermis. And if you dissociated the entire whole skin, the epidermis, it's quite hard for the enzyme to penetrate. And there's a way of kind of like peeling off the epidermis from the dermis using very short displaced treatment, which is what we did. And then subsequently dissociated the epidermis and dermis separately. So a little bit of like, know-how of your biological system is very helpful. And again, we use these contiguous FACS gates, which allows you to capture everything. But then when it comes to this rare fraction here, the CD45 positive DR negative, and you can even split it further, which means that a cell that would have otherwise been present, 1 in 1,000, so it means for every 1,000 cells, you get 1 of it. You can easily make it to be represented at 100 cells for every 1,000 cells you analyze. So it allows you to kind of like be able to pull them out, because you give the clustering algorithm more power by having more cells of the same type to allow this to happen. And we use this sort of like very similar strategy for the epidermis and dermis. And the whole idea here is to cells and downsample abundant cells, because otherwise if a cell is 999 out of 1,000 cells, that's all you keep getting. So you're sequencing a lot of very similar cells. So there are tricks that you can play to kind of like allow you to upsample and downsample. And by doing this, in our skin data set, we've got innate lymphoid cells and natural killer cells. And these are super rare. And so, it's just knowing how to kind of like enrich for them and including them as a more representative population in the analysis to be able to understand them a bit better. I've just put this slide because I think some of the value of the single cell data sets is to be able to then look at the expression of genes that are known to cause diseases. So in this case, specific skin diseases, and you can see which cell types express them, and really trying to bring that to the clinic and making it far more useful, because when a clinician sees the patient, they'll be better able to understand why the clinical phenotype of the disease is manifesting this way, and perhaps for some of the known diseases, things that they may be wanting to investigate and look for because of, the gene expression profile tells you that some other cells would also be affected. So when we've all had a terrible, challenging year with COVID-19, and when we entered that, when we went into the pandemic, what we felt with the HCA was we had this infrastructure research framework and this team science and a lot of know-how of how to apply single-cell multi-omics to now understand disease, and we pivoted our attention to COVID-19. And this is one of the ways that you can pull together resources and be able to sort of like enhance the power of your analysis. So this is a collaboration that spanned Newcastle, Cambridge, and London. And and naturally it involves lots and lots of individuals, those who are at the interface with the patients to kind of like help obtain the material and all of the kind of regulations that are involved, and those who are involved in the data generation, the data analysis, the data interpretation. And these were the junior researchers, and you can see the number of individuals who were involved in the data generation and also in the data analysis. And this is a manuscript that was published a week ago on the immune response to COVID-19. And I just want to kind of like highlight some of the advantages of this approach. So I talked about multi-omics measurements. So here we analyzed 130 patients with varying severity of COVID-19, and we did the whole transcriptome profile. We analyzed the T-cell receptor VDJ expression profile, same for the B-cell receptor, and also the surface protein using the total seq panel, which had 188 proteins. And this really came about because most of us started talking at the start, before we undertook our experiments, how we could synergize and complement. And we decided to sort of like use the same protocols and try to standardize how we would undertake the work, and also the type of patient cohort that we would analyze. And this was a cross-sectional survey. Patients came to the hospital, they got blood, and it would be their severity assignment at that point. So then you can use this data set from peripheral blood, and I'm just putting this up here because the data is available to browse and also to download. And what it allowed us to do was to use the information by analyzing the peripheral blood cells as a window to understanding what is happening in the tissue where the infection, the primary infection is set, and also how the body's systemic response, including the bone marrow, responds to the infection. So these are some of the ways that you can then extract that information and to better understand the response. So one of the things we did, as I mentioned, we looked at RNA, protein, and TCR, and this was super helpful for the T-cell analysis because, cytokines, for example, in this case IL-22 or IL-17, they're much better detected by RNA. But you also have surface protein, for example, CD45RA and RO expression, which allows you to much better identify naive and memory cells. And then the TCR information not only allows you to look at what cells are clonally expanded, but also which cells are actually T cells and which cells are not T cells, i.e. innate lymphoid cells. And the same for B cells. Not only could we identify all of the different types of B cells, including switched memory and non-switched memory B cells, we could identify whether they were producing IgG, IgA, IgM. And this was really helpful information because one of the observations we made between healthy and asymptomatic and symptomatic individuals is that in asymptomatic individuals, there's the preservation of the mucosal IgA and IgA2 responses. So all of this information that you can get from the simultaneous measurement of RNA, protein, and TCR and BCR. And all of this data set is available, and it's there on covid19cellatlas.org. If you click on patient donors, you'll be able to see all the different studies and the data from our studies here as an object for people to analyze and also to browse on the web portal that we've provided. And this sort of like integrated information of studying all the different immune cells allows you to understand the coordinated response, what's happening at in the myeloid cells, what's happening in the progenitors and how they may be in a circuit with the myeloid cells, and the contribution of the peripheral blood cells to the peripheral tissue, such as the alveolar macrophages, and the adaptive cells in terms of the T cells and plasmablasts and plasma cells in patients with asymptomatic disease and severe disease. So, you know, it's not to dwell on the biology so much necessarily now, but just how this type of approaches can allow us to understand. So I'm going to stop here. My lab to thank for this fantastic journey. Lots of collaborators, funders, and a lot of the program or the work that I've described here is undertaken together with the Single-Cell Genetics program, which is led by Sarah Teichmann. And I'm going to stop sharing now and very happy to take questions.
Thank you so much, Mustafa. That was a great talk, and your the final data that you were showing regarding COVID.
Oh, did the slides change? I've just seen the chat.
It did.
Ah.
And they were just worried at some point because you were talking a lot on the same slide, so they were like asking.
Oh, okay.
It was an issue, but no, it was great. It was all great, and it's really great to see the COVID, the results on COVID. It shows how people have so much infrastructure and so much collaborating power nowadays that they can generate so much data so fast and help each other in the process, right? So that was really great too. We have a few questions here from the audience, so from the attendees. The first one from Raul Arias Carrasco. Are there HCA-specific validated protocols for tissue processing in each biological network?
I think there's quite a lot on protocols.io and also accompanying the publications, and that's worth looking at as a resource. And I think fundamentally, you know, there are considerations for tissue processing in terms of like what type of questions you want to answer, what type of cells you want to capture, and yeah, that's worth thinking about. But protocols.io and the publications are available. I don't think I can speak for the developmental biological network. I don't think we've put aside a specific within the biological network specific protocols, but that's something to think about. Thank you.
Okay. From Sanhita Banerjee, how do you detect the increasing number or accumulation of a particular cell type from healthy to disease states when the expression level of signature genes remains the same in both conditions?
Yeah, so there are several ways of doing this. You can kind of like look at it as a proportional increase of or change in those cells. So if you were to take all of the cells that you analyze and then look at the proportional upregulation or downregulation, it may be that the gene expression profile is the same, but the proportion that they're present will be different. And you can do that by taking into account of, you know, say, for example, immune cells, monocytes. So what proportion of CD45 positive cells were monocytes in health, and what proportion of CD45 positive monocytes were in a particular disease state? You can also quantify this with absolute numbers. So if you could, you know, in the case of peripheral blood, for example, which is a good example, you can do sort of like a true count, like, you know, the way that full blood counts are done, where it gives you like number of cells per volume of blood. And then if you've got that measurement and with your analysis, you can then correlate and actually show number of cells per volume of blood. So those are the ways of doing it.
Okay, great. Still more or less related to that, from Luis Felipe Antunes Branco, great presentation. Focusing in skin cells, what would be the approach to investigate different states of the same cell and for interactions between these cells and the immune system cells in skin diseases?
Yeah, that's a good question. So there are two ways of looking at it. So you could take, one way of doing this is if you were to just kind of like cluster and annotate the cells where all of the cells are present, you may not be able to get some of the resolution. So you can then subset the data. So for example, in skin, you can take, stromal cells, or you can just take them, antigen presenting cells, subset that bit of the data, and then recluster, and then sort of like zoom in. So it's a bit like, you go zoom in, zoom in, zoom in, and then you can, look for more heterogeneity within the same cell. But this also calls for some level of interpretation as to what level of kind of like splitting it makes sense, because otherwise you just sort of like you over cluster, but the cluster, the differences are no longer biologically meaningful. So that's one way of looking at different states of the same cell. And then you can also take a state where it's steady state or health and look at it in a perturbed state or in disease. So that's two ways of looking at the states of the same cell. And then for interactions between these cells and the immune system in the skin diseases, if you were to just use suspension data sets, as I said, many of these kind of like receptor ligand interactions can be used to infer interactions, and therefore how stromal cells or endothelial cells may be interacting with macrophages. So in our manuscript, we describe an interaction between vascular endothelial cell and macrophage type, which is involved in what we, what we propose as recruiting immune cells into the skin. And you can read more into it's in Reynolds et al., which we published in Science earlier this year. So that's one way of going about it. And then based on those predictions, you can then look at the surface protein expression and also visually or spatially analyze the tissue in situ to show that the cells indeed are, next to one another and express the receptor and ligand. So that's kind of like adding to the validation. Or if you have a system that you can perturb, you can either block that interaction and so on and so forth to see how it then changes. So those are the ways that you can do it.
There is another question here addressing the same topic, because it talks about granularity and how wet lab biologists should discuss with computational biologists about that. That's from Victor Aliaga Tobar. Says, hello, very nice presentation. With the advent of these technologies, we have acquired an enhanced ability to phenotype cells and cell states, in some sense developing depending on the choices of experimental approaches you have discussed, phenotyping would have varying degrees of granularity. How to reconcile this a priori decisions with the further analysis and discussions of states? What information in this regard should the wet lab biologists discuss with the computational biologists previous to the analysis to avoid overstatement of results?
I think that's a very important, almost point and question. I see the way we go about this as very much a teamwork in that, involve the computational people from the very onset and, outline what are the research questions that you want to get, what are the kind of like known a prioris about that field, and then seeing what the data shows and then interpreting together the data sets. So it's very much a partnership, but a partnership that's done at the very onset, rather than you generate the data and then you get the computational person and say, can you analyze my data? It's much better that way. And having a lot of experts in the particular field, when I kind of generated all that, when my team generated the fetal liver data, we didn't really know much about, hematopoietic stem cell progenitors or erythroid differentiation or some other lineages. So, we had different collaborators who are experts in those areas. And with the COVID-19 as well, the one reason we could move so quickly was, we had experts in progenitors, we had experts in, mononuclear phagocytes, we had experts on T cells, and we had experts on B cells. We did a divide and conquer, but always the discussion was together with the computational person. And yeah, it's very important to avoid overstatement of the results. And there's it's not an absolute science. I think people kind of like expect the data, the transcriptome data to just say, this is cell A. It isn't like that. It isn't an absolute thing. There's a lot of interpretation and discussion back and forth as to what is biologically meaningful and statistically robust.
That was a perfect answer. Regarding a little bit that too, Carla Vargas asks about COVID patients. She says, thanks for the presentation. I'm curious about the samples from the COVID patients. Can you tell us more about that? Also, does the results change drastically from fresh to frozen tissues?
Oh, okay. So the samples from the COVID patients were basically peripheral blood. So the patients came into hospital, they had their blood taken, and their severities at the time when the blood was taken. So I had it earlier on about the different disease severities. Either they were healthcare workers who were asymptomatic, or those who were asymptomatic had symptoms, didn't require oxygen, had symptoms, needed oxygen, had symptoms and were in ICU and required non-invasive ventilation, and those who were in ICU, intubated and ventilated. The samples, we froze them down. We basically did lymphoprep step and then took the peripheral blood mononuclear cells, froze them down before we use. And whether the results change drastically from fresh versus frozen tissues, I can tell for blood that it doesn't really make that much of a difference. But plasmacytoid disease are a bit more susceptible to sort of like loss from freezing. But if you freeze it down kind of like carefully, it's okay. I can't tell very much about all the different types of tissue, whether there is a difference between fresh and frozen, because it depends on the composition of the tissue and which cell types are more susceptible. Certainly, I think hepatocytes are more susceptible to cell death than immune cells. And the other thing to consider is therefore, you could use nuclei sequencing, for example, for frozen tissues, because, the cell, you may lose the cell, but you will still have the nuclei, which you can analyze, but also dissociated tissue versus nuclear extraction and nuclei sequencing enriches, or you're more likely to analyze, you're better at picking up epithelial cells from nuclei sequencing, whereas you're less likely to lose more of the epithelial cells when you do kind of like tissue dissociation or RNA sequencing after freezing the cells.
I think that goes also back to one of the questions talking about the choices of experimental designs and how you should discuss that, because that's all related to the results you're gonna get, right?
Yeah.
There's a final question from Swanti Gupta. Can we know that we're developing, can we know that we're developing in a certain process, say neurodevelopment, how do the gene transcript abundance change as clustering is only on basis of surface markers?
I think most of the time for RNA-based analysis, it's not on surface markers, but or the entire transcriptome of the cell, which includes both surface transcripts that encode surface markers and also intracellular markers.
So thank you so much, Muslifa, for being here with us today. It was really great to have you. It was a very, very complete talk. Helped a lot of us here who have not had access to this kind of very detailed information before. And we hope to have you back soon.
Thank you very much.
Thank you. Thank you. Bye. So with this, we concluded the morning sessions.
Let's begin the afternoon session. I'll introduce myself first. My name is Ignacio Wichmann. I'm an assistant professor at the Department of Obstetrics at Universidad Católica de Chile, and I'll be moderating these afternoon sessions. Okay. So next we have Enrique Sapena Ventura, who studied biology in the University of Valencia in Spain. And after that, he did 2 master courses in virology and bioinformatics. Now, he joined the EBI shortly after finishing the 2nd master as a data wrangler for the Human Cell Atlas. Since then, he has been working in bringing high-quality metadata standards for the community, engaging in scientific events to ensure that the data coordination platform stays in touch with the fast-evolving single-cell community. So, with that said, it's a pleasure to have you here, Enrique, and we look forward to your upcoming talk.
Thank you, Ignacio. So I'll begin sharing my screen. So basically, I was supposed to go first in the today. I swapped my spot. So I am going to take a little bit of a step back from the first and second talk, and I am going to begin talking about public repositories for single-cell genomics and transcriptomics. So as Ignacio has said, so I work at the MBL-ABI. One of the departments here is the AIT, which is the archival and infrastructure team, and they are responsible for bringing the databases, the archives that are behind this that the scientists look at, and keeping them in touch with the community. So first, I am going to start with a little bit of history on data sharing. And then I'll talk a little bit about the INSDC. Don't worry about the acronym. I'll explain it in a bit. And then I'll show a little bit of other platforms for genomic and transcriptomic data. And I'll focus, of course, in the HCA because I'm a little bit biased here. I work for them, but and also there's not enough time for explaining everything. And then I'll make a little insights on submitting your data, which is an important part of the process, and an honorary mention to portals. So let's start. So the first question that pops to my mind when I think when I think about all of this is, so what is good science? So good science, one could argue a lot of things about what good science is, right? It's been talked, it is still talk, and it will be talked about. And but so you could argue that good science should be reproducible, right? Like one laboratory in one part of the world should do an experiment, and other laboratory, if they reproduce the experiment, they should get the same results, right? And the list goes on and on about all the qualities that good science should have. But today I'm going to focus on one of the qualities I think that makes science, that makes good science, which is shared science. So basically, sharing ideas and data, there are studies around that have shown that sharing these ideas and data impacts the outreach of those publications, and make them so that they can reach a bigger part of the community and make a better impact in future research. And this data sharing has always happened, whether it has been, for example, at the pub, or through letters, or in the recent days, usually through internet. So in the recently, we have developed a lot of technologies, the technology of information, right, which has led to a lot of opportunities, but also a lot of challenges associated with this easiness in sharing data. And even more recently, there has been a big paradigm change in the scene, which has been the big data, not only in biology, but in many other areas of research. So one example of this is in the 90s, with the start of the Human Genome Project. There was a big increase in data. It wasn't anymore about just genes and studying a specific variation of 1-2 genes, or maybe 10-20 genes. They were talking about sequencing the whole human genome. And it was a very big and ambitious project. It was successfully executed. But it brought with it a lot of issues that needed to be solved, right? Like, how do we share the data, for example? how do we share this big apparatus of data? And of course, setting up the standards to share that data. If different laboratories across the world were working on the same thing and they were trying to complement each other research, it would prove very difficult to complement that research if the standards were not the same. I have pointed here to a very interesting article, but I hope to share my slides at the end, so you will be able to look at it. And so yeah, so basically with this in mind, how were these issues tackled? So around starting the decade of the 80s, there were several conversations and several meetings across the globe from very different number of researchers, like names, and they were discussing about how to tackle this. And by around 1987, the INSDC was born, the International Nucleotide Sequence Database Collaboration, which was a celebrated global initiative, and it is going even to today, and it has a really big impact. And it's a collaboration between three main entities. These three main entities being located across the world and being the DNA Data Bank of Japan, the NCBI, I'm sure you've heard of it already, in USA, and ENA-EBI in Europe. So the main goal of this collaboration was to share not only to share the data, but to make it accessible. And in order to share the data in a good manner, they planned to set up standards, formats, and rules, and thus avoiding duplication of effort. So this is all good. And as I said at the very beginning, I am going to focus only on the ENA. Unfortunately, I did not have enough time, physical time in this presentation to talk about everything, but I'm going to introduce you to ENA and a little bit on how to explore it and what should you expect in there. So if you think about this, so the first question that pops to mind is, how do I find the data? But before that, before you try to find the data, you need to understand what data is in this repository that you can find, right? So basically, the data in ENA is presented in hierarchical tiers. The first one being the reads, of course, which represent biological reads and are organized in random experiments, which represent the sequencing data files and the sequencing methods. Researchers can also upload assemblies like whole genome context sets, transcriptome sets, scaffolding sequences, and even fully assembled chromosomes. And then on the last step of the tier, you got the annotations for coding sequences and non-coding sequences. And all of this is encompassed by a project, and this is more or less the overall layout of a project. Do not worry too much about memorizing this. It is very intuitive, and we're going to go through it later in this talk. But what you need to take from this slide is that you got a study. This study contains experiments and runs, and these experiments and runs link to samples, which contain the metadata for the biological material that resulted in these sequence files. So basically, I am going to explain now how to find the data. They have many ways to find the data, but I'm going to focus on the user interface. You have many, even in the user interface, you have many ways of searching for the data. So basically, I am going to focus on this, and I'm going to go one by one, explaining what you can do with this to optimize your search. So the first option is the free text search, and it's basically what it is what it is, right? It's free text search. It's the only thing that I want to point here is that you can link terms with syntax. So for example, there is a lot of documentation on this syntax, and you can look at it. But in this specific example, if you search for kidney and cancer and single cell in quotation marks, you will find only the projects and sequences that contain the word kidney, contain the word cancer, and contain the phrase single cell, like that, without any variation. So the next, but if you don't want to learn how to create these queries, which is understandable, there are a lot of parameters and it's not always super intuitive for non-computational people, they have this option of advanced search, and in which you have all these options. And the first, I'm going to go one by one, so it's going to be breaking down pretty easily. Then the first one, you choose the type of data return. For single cell transcriptomic and genomics data, you want to aim for studies used for raw reads, and you can see that there is another option here. Use a previously defined rule-based search. Do not worry too much about this. You can use it. This is basically after you have created your search, you can save it and you can share it, just in case you want to share your search with other colleges you're working with, and you don't want to tell them everything that they have to fill in to find the search that you put. So then after that, you have a very nice, very nice, I am a data curator, so I have been through several search interfaces, and I have never seen anything like this to build queries. It is very nice. It gives you a lot of flexibility and it helps you a lot. So for example, here, I am able to click on taxonomy and related, choose the database by which it will look the taxonomy. And if I type Homo sapiens, I will get a list of results, of course, in this case, only one. And if I put that and update the query, the query will appear. And this is a very powerful tool to create queries that are very specific to your needs. Then after that, after you have created your query, you can include or exclude accessions. You may want to share a search. You may want to include in your search things that do not have specifically the terms that you are looking for, but you think are interesting for it. So you can include them here, and also that you have the ability to exclude them, right? So the next two ones is to choose the return fields. As you can see, the list, as you can see by the size of this, the list is very big. So I have not gone through, I'm not going to go through all the fields that you can return, but basically you can choose from here. You can look at it yourself and choose the fields that you want to return. And of course, just to finish, you can just escape or limit the number of return records. So that is the advanced search, and like you hit search and you will get all the projects that match your very specific and advanced query. And lastly, I'm going to talk a little bit about the cross references. So basically, as you may or may not know, the databases are connected to each other. We are not competing against another. This is about sharing. So the ENA has a very nice tool to cross-reference with other archives, and like you can also search by ENA record. It's pretty self-explanatory. If you have, like, for example, an array express accession, you can link it there and search for it in search for the data in ENA. So let's say that now you have found the project that you want. It may be more than one, but for time purposes, I'm going to focus only on one. So now, not what. Now, what is the next step? So now you click on the project, and you are presented with a lot of information at once. So when you click in the project, you will get this page, which is basically like a general description of the data set and links to important information. And you'll be given an accession, which is a unique identifier for the project, and it's the thing that you will always find in the papers, right? Because with this accession, you can know that you will only point to this data and this data alone. So I am going to focus on this part of the web page, which basically shows you like all the different possibilities of what you can do with what else you can show from here. So I have written a little bit of a summary here. Basically, the view and download is just view and download an XML version of the metadata if you are not used to it. XML is a flat text file format. It's very much used in a lot of APIs. But yeah, you don't need to worry about it too much. And the read files, which show a table with varying degrees of information for sequencing files. You have also additional attributes, which is basically additional metadata about the submission. That sometimes you may get also parent projects, because one project may be associated with a super project. Let's say they call it umbrella project. And you have another option to show the related DNA records. So you can get a list of all the runs and experiments that are within this project. So I am going to focus for the purpose of single-cell genomics and transcriptomics. I am going to focus on the read file part of the web page. So basically, when you click on that, you are shown this table. And what is this table? What are the rows? What are the columns? So basically, each row shows the corresponds to one run, and each column, in each column you have specific information about that run. So, for example, you have many fields available by default. You have accessions, you have the taxon, so in this case, Homo sapiens 9606. You have FASTQ information and then low link. You have other file downloads. For example, sometimes instead of FASTQ, you will find BAMs or process files. And you also have the SRA file download. So as you know, as I have said before, these databases are they share data, so you can also download the SRA from ENA, and you can get also other information. I am not going to go through too much detail, but if you click on Show Column Selection, you're presented with this very small table with almost no fields. But basically, I think what it is a lot of the times important is the fields with FASTQ prefix, which shows the information about the FASTQ files, fields that have the alias provided by the researchers when they deposited the data. Do not underestimate the power of the aliases that researchers give. They are usually very intuitive, and they can tell you a lot about the sample. And finally, the library strategy, the library layout, and the library source. For these three, there are accepted values. So they are all linked on the read the docs documentation of ENA. But basically, they will tell you about the strategy for the library preparation, the layout being a pair of simple, and the library source being, for example, Transcriptomics or Genomics, etc., etc. If you are interested, I suggest you go to the ENA read the docs. It's very interesting, and yeah, and they have a lot of information there. I had to sum up a lot. So the next step, once you know what you're looking at, is how do I download it? You need it locally or in a server in order to process it, right? So from here, I just want to point out that you can download two types of things that are related but not the same, which is the data, which may be the sequencing files. They are either raw sequencing files or process, like matrices, for example, and the metadata, which is information about the experiment. So for the information about the experiment, you can either download the XML reports. If you are not experienced with XML, I wouldn't recommend it too much. It's hard to parse for inexperienced users, but if you are interested, take a look at it. And there are Python libraries to translate this XML information. But what I think it's more interesting for the researchers here is downloading the JSON and the TSV reports. So basically, you can download this table with the return fields that you want in any of those two formats. I personally prefer JSON, but I understand that this is not a format that is widely used outside of the bioinformatics. Well, bioinformatics, but it's hard to parse for people with little experience, computationally speaking. So you can also download a TSV, which can be loaded into Excel, and it's easily readable, and it will, as I said, contain all the information. And about the data, they make it super easy in ENA. You just have to check the boxes and click on download selected files. That is the way you do it with the user interface, of course. There are many other options. Again, in the ENA read the docs webpage, you can find all the options. There are many from FTP downloaders to apps like Globus to Python packages. You can even download the metadata and use wget to get the files from the URLs. You can access all this data from the REST API. The options are varied, and I have to say that I have tried a couple of these, and it's really nice. So if you are really interested, I suggest you to take a look at it. So I have talked about ENA, but I think a lot of people have seen that there is not only ENA, SRA, and DDBJ. There are other platforms to look for data. A quick insight here is that the INSDC, its aim to be the main source of truth for the data. So whereas you may get other platforms to that can be very specific, like for example, the Human Cell Atlas can be very specific for human data, and we try to focus on healthy data as well. The INSDC is supposed to be the source of truth from which the other platforms can take that data and present it and curate it maybe to their standards. So you see, I have put three examples here: Human Cell Atlas, ArrayExpress, and Geo, the Gene Express in Omnibus. But there are many other more, and I realized that I do not have much time, so I'm going to focus on the Human Cell Atlas. A little bit biased here, maybe, but so again, what should I expect to find in the Human Cell Atlas? So here, this is a lot, but don't worry, we're going to go step by step. This is basically an overview picture of how a project, a data set, is represented in the Human Cell Atlas. So for here, we try to represent the experimental structure. So here, for example, you have a donor organism. We can call her Kate, the doctors took a biopsy of her, and they got a pancreas sample, a tissue sample, block of tissue, or just a small tissue sample, and through an enzymatic protocol, they dissociated this sample into a cell suspension, and this cell suspension, when as others have explained before me, they went through the library preparation methods and sequencing, like maybe with 10x, maybe with any other technology, and generated the FASTQ files, and from there all the downstream analysis. So you can access the data in this web page. It's very intuitive, very nice. And here in this page, you can see that you have an option to search by filters, and you can filter by actually a lot of metadata. The HCA is a highly curated platform, so you can use the organ, technology, type of tissue, the files that you want. Maybe you only want BAM, maybe you want the FASTQ. So you can select all of that in filter projects, and you can also show select here projects, samples, or files, depending on what you want to see. So this is an example of what I was saying. Basically, like here, you can see that when we click on tissue type, we get a lot of information that we can filter by. We can filter by organ, like one tissue, for example, like selected cell types, if we are interested in a specific cell type, etc., etc. I recommend you take a look at it and just play with it. Don't worry, you're not gonna break anything. So just try to make it so that it fits your research. And as we can see here, we get our query here, and instead of like the 55 projects that are currently in the DCP, it will show only the projects that fit this query. And if you click in one of these projects, you will get a project overview. There is a lot of information to unpack here, but basically it's general description and some summary metadata. If you click on the top left corner on the project metadata, you will be presented with this web page in which you can download the TSV with the metadata. And what is this TSV? So as before, it is a top-separated text file that contains all the metadata for this project. In this case, it is a little bit more packed than the ENA one. I am just showing a very amplified part of this TSV, but basically the information, the main takeaway that you have to get from here is that there is like one row is one file. The header contains the programmatic names of the fields that are in those columns, and that the same they are linked by entity document ID. So for example, if you want all the files that pertain to a specific donor, you have to locate that donor's document ID, or if you're interested in cell suspension, same. So basically, about the about the programmatic names, we have a metadata dictionary in which you can basically take a look at it. There is a lot of information, but a very useful point to take here is that you can search the programmatic names, and it will return it with a description. I have an example here actually. Let me just show you. So I have searched for read index, and it shows me the field, the full name, and a description of the property. So let me go back to the presentation. So yeah. So basically, continuing with this, if we go to the next tab, we will find the project matrices. In this case, we've got two types of matrices. We have the DCP generated matrices by the standard pipelines of the HCA, but they are count matrices. They are not normalized. So you need they need some work afterwards to be analyzed. Contrary to the contributor generated matrices, which are usually fully analyzed, fully corrected matrices that are provided by each contributor, because we want to also share the expertise of these contributors. We want to standardize the output of data, but we also want to share the expertise. So, and lastly, you get like a final final page with some external resources like links to the archive data, repository links in case there is any code that they want to that the contributor wants to share, analysis portals if there are any. For example, like it could be linked in the single cell expression atlas, for example. Yeah, and just super quick, because this part is actually super easy. I'm just going to show you how to download the data and metadata. The metadata is a click. You can either go to the web page I showed before, or in the main page, you can click on each project metadata. And for the data, you mark the checkboxes for the project and click on export select data. And once there, you can use any of the options to download either locally or to or export to a cloud-based platform. And you can also the files that you can download are FASTQ, DCP produced BAMs, if available, and the matrices. So yeah, that is basically the HCA. So I wanted, before I finish the presentation, I wanted to give an honorary mention to portals. They are not exactly repositories for the sequencing data on itself, this raw sequencing data, but they can host and show the process data, which is very, very valuable. And there will be many times in which the data cannot be shared publicly. Like, for example, in the great example that must show with the COVID-19 cell atlas, a lot of the data in there are matrices, because of course patient information is very sensitive and have to be treated. It has a lot of privacy concerns. So yeah, and I just wanted to give an honorary mention to these ones, and of course the COVID-19 cell atlas, which was a big overlook that must just remind me about. And yeah, and just so you'll know, these archives are not black boxes. You can submit your data to all the repositories, and they all have their user interface submission and like a programmatic submission, and they have support, and they will help you get your data there. And they are usually very wonderful people. I know all the ENA team, They are wonderful people. I have worked very closely with them. And just as a little bit of a plugin, if you have some data, some human single cell data, you can contact the Wrangler team at the Human Cell Atlas, and we will help you get your data both into the data portal and ENA. So basically, what we have learned today is a little bit of the history and context of data sharing. I talk a little bit about the history of data of the INSDC repositories, but I focus especially on how to search, interpret, and download data from ENA. And I wanted to, I spoke a little bit as well about the HCA and other platforms. What are they? And how to submit your data. And didn't already mention the portals. So lastly, I want to thanks to all the wonderful teams that I work with, not only as a member of the MBL-EBI, but as a member of the Human Cell Atlas Data Coordination Platform, and to the whole data coordination platform team. They are wonderful, and they are working very hard to make all this, make a very nice interface and resource for researchers to share their data. So thanks for listening. Are there any questions? Stop sharing now.
Okay, thank you, Enrique, for a very didactical talk. It's like watching a tutorial. Very, very nice. So we have a couple of questions from the audience. Tiago Lubiana asks, well, he first says that nice service. He asks, are there any plans to support semantic queries, for example, via SparkQL? It would be nice to find datasets using some ontology terms.
So I am not entirely sure if there are plans to support semantic queries. I do know that there are already some examples of services at the EBI that are implementing ontology expansion in their queries. So, for example, biostudies. If you search for a term just like kidney, it will search for the parent terms of kidney. So yeah, I'm sure there are some plans. So I work closely with them, but I do not know all their roadmap. But I'm sure there are plans because I have already seen some examples of it being implemented in some services.
Okay, so we have another question by Samantha Kuwada. She asks, what are the main differences of ENA and dbGaP SRA repositories? It is well discussed, the difficulty to work with SRA dbGaP data, since there isn't a consensus of data format. For example, every time that you submit a data requirement, it takes a long time to understand how the data is. And even though there is a guideline regarding how data should be uploaded, it is not always followed. Any comment regarding data submission in this repository specifically?
Yeah, that is a very good question. So first of all, the ENA and the SRA, there are some differences in implementation, but there are not that many because they are supposed to be mirrors of each other. In the case of dbGaP, this is for managed access data, so it's a little bit different. But yeah, I do agree that sometimes it is very difficult to understand the data because these kind of services, they get a lot of submissions every day. So even they try to have a very uniform and understandable format, but it's hard to, I will say that it's hard to work with SRA data in a programmatic and standard way, but it is as well with ENA because of the things that you mentioned, like sometimes the guidelines are not followed, and yeah, there's not much you can do about it. I would suggest like you won't find the same amount of information, but there are other platforms with the same data, with part of the same data, but more curated, like ArrayExpress, for example, and the Human Cell Atlas. We don't have many data sets right now. We are looking towards incrementing this number, but yeah, there are curated, openly public databases that you may find a data set of interest in there, and it will usually be easier to understand because they have data curators working on those data sets.
Okay, so very much related to this question, who curates the data for the HCA, for the Human Cell Atlas?
Again, sorry?
Who curates the data for the Human Cell Atlas?
Well, that is a very good question. I do, for example. So in the HCA, we have a data wrangler team, which is basically, it's a fancy term for data curator. I always tell people this. So we are not only at EBI, but also at UCSC. UCSC has their own team of wranglers. And we find interesting data sets, and we, based on the paper and in our own metadata schema, we curate the data and metadata, and we try to like be consistent, because it's not only about like having the same fields, like the organ field will always equal organ, but also like there are some difficult examples in which we've had complicated discussions to arrive to some kind of agreement. Like, for example, for bone marrow, what do you use? Do you put bone marrow as an organ, blood as an organ? How does it relate to other, So yeah, we the Wrangler team is the one, are the ones that curate the data, and you can. I have the email in the slides. I would like to share the slides at the end of this talk. It's a Google Slides,
Okay.
so everyone should be able to access, and the email is there. And yeah, feel free to drop questions in there. We are very happy to answer.
Okay, that's very nice. We have two questions from Benjamin Pizarro that are related. He asks, are there any APIs to access the data directly? And also, is the backend of these queries in a specific language like SQL?
So to the first question, yes, there are REST APIs to access the data. Yeah, I have not focused much in the APIs. So the backend of these queries are, it's basically REST APIs, yeah. So it's basically sending queries through sending, yeah. It's not as yeah, sorry. It's not SQL. It's just as far as I know, they only have the REST APIs exposed. But you can access, and like there is a lot of information in the read the docs. Let me just 1 second. So I am going to share a link through here and to all attendees. So this is the Read the Docs webpage. If you are interested in looking into the depths of what you can do programmatically, I suggest you begin looking there. They have a lot of documentation, lots of.
Okay, perfect. Can you also post the link to the slides on the chat?
Yeah.
So that everyone can have access to it.
Mm-hmm. Sure.
And I was wondering, another question while you type that in. You mentioned that many of the data sets aren't available as raw data because of data protection and patient ID protection, and it's a very delicate matter, right? Mm-hmm. So when you're working with already processed data, you'll also have to work with the pipelines that they use to generate it, and that will sometimes just potentiate the potential batch effects that you can have between one data set and the other would you have any comments on that, on how to integrate maybe working with multiple process data sets?
Yeah, unfortunately, we have not yet had managed access datasets in the HCA. It's in the DCP. It's in the roadmap. So we plan to have our first batch of managed access datasets very soon, but we have not yet worked with managed access data and with batch the batch effect that comes from having data processed from multiple sources, multiple laboratories.
Okay. Okay, so thank you very much, Enrique. That was a very nice talk and very nice Q&A session. So
thank you very much.
Thank you. I think that's all the questions we have for now. So next up, we'll be talking with Raza Elmentaite. So I hope that I pronounced that correctly, Raza. Okay, thank you. That's good to know. Raza has completed an integrated bachelor's degree and master's degree in molecular and cellular biology with biotechnology at the University of Glasgow. Now, she's a Wellcome Trust Sanger Institute PhD student supervised by Sarah Teichmann. Okay, so Raza has a wet lab background and an interest in computational single-cell RNA-seq analysis. As part of her PhD project, she is currently working on deciphering developmental dynamics and cell fate decisions in the human gut. So, Raza, it's an honor to have you here, and please, it's all yours.
Can you see the slides okay?
Perfect.
Awesome. So thank you very much for the introduction and inviting me to speak today. I'm very excited to present my main PhD project in this workshop. So just to begin with, I want to introduce my team. So I work closely with Sarah. So Sarah is the leader of these projects, and we also work closely with Matthias Ilbauer, who's our clinical collaborator at the University of Cambridge and an expert in IBD biology. So that's sort of bringing this interest in the gut biology and combining with Sarah's interest in single cell. And I also work closely with Kylie James, who was a postdoc at Sarah's group, but now has moved on to a group leader position in Sydney, and also with Alexander Ross, who's a clinical fellow at the University of Cambridge. And really, as a group, our goal is to contribute in creating a comprehensive survey of intestinal cells in the human body, and hopefully discover some interesting biology along the way. I think it's a hope for everyone in this workshop. Just to mention that the data that we generate, we usually upload to HCA, and we also share it via ArrayExpress, but we also have our website, gutcellatlas.org, where you can quickly use an interactive viewer to look up the gene you're interested in. So this is something that Enrique was telling about, the CellX gene interactive viewers. So this website is based on that. So why are we interested in the gut? So the gut is really highly structured organ that has so many functions in different regions. And what we're particularly interested in is that the lineages and their activation potential really changes throughout development and adulthood and during the disease. And to add on that, there's also constant exposure to varying environmental factors, such as colonization with microbiome and activation of different immune populations through that. And so, yeah, our goal is to really comprehensively map the gut. So just to mention some previous work that we've done in the lab, this is a paper that has been led by Kylie, and where she used single cell sequencing to sample different regions of human colon, so cecum, transverse, and sigmoid colon, and also simultaneously shotgun sequence the bacterial species in these regions. And what you could observe is that there's an increased activation and clonal expansion of immune cells in the regions where there's more diversity of microbial species. Sort of like one of these first studies where we try to combine single cell with microbiome and better understand the immune system in the gut. The second work that has been published last year was led by me and Alexander, and here we focused on profiling the cells during the intestinal development and also in pediatric healthy and diseased donors. And the process that we focus there on is this formation of cryptand villi structures in humans. So basically the folding of the epithelium in the gut, and how different lineages, for example, fibroblasts and smooth muscle cells, influence this process during development. Just following up on that, sort of that's where our project comes in, where we are trying to create this more comprehensive view of gut throughout different stages of life. And so in addition to the first trimester samples mentioned before and pediatric samples, we now also sampled second trimester developmental samples with help from Human Developmental Biology Resource in Newcastle. And we also were lucky to get samples from the adults, so from organ donors 20-75 years old. And where possible, we try to get not only gene expression, but also BCR and TCR sequencing, from which we could infer the immune cell clonal expansions in these samples. But in addition to this time sort of axis, we also wanted to have the space axis. So we wanted to also sample different regions of the gut because it's so diverse and has so many different functions. So in development, we've sampled proximal ileum, which will develop into duodenum jejunum regions, then middle ileum, terminal ileum, and colon as a whole, as in that time point, colon is actually quite undifferentiated, not yet having the regions that you would see in the adult. And we were also able to retrieve the developing mesenteric lymph nodes from samples 15 weeks upwards. So that's quite a unique aspect of this data set. And from the adult samples, Kurosh, who is a surgeon at the University of Cambridge, has done, and his team has done an amazing effort in sampling up to 11 different regions of the adult gut. And we've sampled duodenum, jejunum, terminal ileum, appendix, cecum, ascending, transverse, descending, sigmoid colon, rectum, and mesenteric lymph nodes. So in total, with all the time points and all these different regions, we had 164 different 10x runs. So that's obviously a huge project. So this is a UMAP of all the different cells that we have captured in this data, and we have over 400,000 cells with 9 different lineages. Just to mention that we use BBKNN integration, and I work mostly with SCAMPy pipelines, well, packages that are available because it's sort of easier to do this large-scale analysis using Python rather than R, but that's everyone's preference. So just sort of, I realize my talk is more about biology, but I hope that's still relevant. So some of the things that we were happy to see in this data set was that, for example, developing mesenteric lymph nodes and adult mesenteric lymph nodes had primarily immune populations, while the other time points where we saw the lineages that we would expect, the epithelium, endothelium, and mesenchymal populations. One thing that we also noticed that neuronal cells were actually mostly enriched in the first and second trimester of development, which possibly is technical sort of, we say, artifact, because it's just harder to dissociate neurons using dissociation protocols, especially in adult samples. But in order to account for some of these technical variability, which is derived from our sampling differences, we apply this Poisson linear mixed model, and this is results from this analysis here at the bottom as a dot plot, where we try to account for technical variability in enrichment of cells at different stages. So in red, you can see the cell types that are enriched, in blue, the ones that are depleted. And this is just a significance level. So the bigger the dot, the more significant enrichment or depletion of the cell type is. And we could see that actually neuronal cells were significantly enriched in the first samples. But again, like, it's hard to say, it's hard for me to say whether that's a biological difference. I'm not sure about that. Anyway, this is an overview of the data set, and I'm just gonna go into some of the topics that I want to sort of discuss in more detail today. So I want to start with epithelial cells and tell you a little bit of insights that we were able to get from this data set. So for those who are not really familiar with the gut biology, the epithelium is actually highly structured in the gut, and there are these crypton villi structures that increase the surface area for the nutrient absorption in the gut. And the epithelium is composed of many different lineages. There's stem cells, absorptive cells, and secretory cells, and many different small lineages like goblet cells, enteroendocrine cells, and tuft cells. So what we wanted to look for in this data set was whether we have any new cell types. Also, if we have any new spatial distribution patterns that have not probably been observed before, and any unique functional insights into known cell types. So I just want to start with this best 4 enterocyte cells that we actually saw some interesting spatial distribution of. So best 4 enterocytes were first described using fluorescence microscopy, and they've been observed in the gut, I think, in 2011. But since then, a couple of groups reported this cell type being transcriptionally distinct in the single cell data sets. So we were quite interested, interested in understanding the distribution of the cell type. So previously, it has been reported only in the adult colon, and the function attributed to this cell type was pH sensing and possibly other ion absorptive processes. But what we could see in our data set was that actually it's present not only in the colonic regions, but also in the small intestinal regions, as well as during fetal development. And we also used Human Protein Atlas to look at the staining of different regions of the gut and confirm this observation. So you can see with the red arrows, I sort of point to these best 4 positive cells in different regions of the human gut. And when we look more closely into this is pediatric and adult epithelial cells with an overlay of SC velo arrows indicating the sort of pseudotime trajectory of these cells. When we look more closely at these best 4 cells, these seem to also sort of create these distinct clusters based on which region they're coming from. So small intestinal ones are in darker green, and the colonic best 4 cells are in lighter green. And we wanted to also retrieve, what's driving this difference. And for that, we used a Milo tool, which I encourage for you to look into. It's by Emma Dan and Saras and Marione's groups. So what we were able to see was that while best4 and some other genes were expressed across cells from different regions of the gut and also different time points of development, there were some other genes that were previously considered as being markers, but were actually more enriched in the large intestines rather than small intestines, and possibly are not necessarily markers maybe that you would want to isolate these cells using. What we also could see was that the small intestinal best4 cells highly expressed CFTR. And that was quite interesting because CFTR encodes for channel, which is frequently mutated in cystic fibrosis patients. And cystic fibrosis patients actually have gastrointestinal symptoms, sort of connecting the function of best4 cells and with disease. The other observation that we actually made using the protein cell atlas was that best4 positive cells were often reoccurring with the goblet cells, these cells that have this blob. They are the cells that produce mucus in the gut. And cystic fibrosis patients actually suffer from defects in mucus production and more frequent infections in the airways. So this sort of created this hypothesis for us whether the best4 cells or this interaction between goblet cells and best4 enterocytes is required for the proper mucus production in the gut. And this is something that we're interested in following up functionally. Another interesting observation that we've done using this data set was in the enteroendocrine cell compartment. And here you can see a UMAP projection of all the different enteroendocrine cells that we find in our data set. And it was quite interesting to see that we have captured this progenitor population that was found in the very first and second trimester of development. And they sort of made this trajectory differentiating in more specific lineages of enteroendocrine cells that they basically are named based on expression of a hormone. So for instance, N cells are the ones that produce neurotensin, a hormone in the gut. So we could see a diversity of enteroendocrine cells, but what was new was that within these enterochromaffin cell compartment, we could see a small population of these cells that were highly expressing neuropeptide Y. And neuropeptide Y is the hormone that controls thirst and hunger in us. So it was quite interesting to see a specific population expressing this, as before using organoid systems, we were not able to visualize a separate, using single cell, we were not able to visualize a separate population expressing this, this hormone. And so to dig deeper into this, we also reconstruct the pseudotime of differentiation from the progenitor to enterochromaffin cells. And these arrows, we point to the factors that have been sort of described before to be important in differentiation of enterochromaffin cells, but also see a lot of different genes that might be important specifically for differentiation to this NPY, NPW positive enterochromaffin cell. Okay, and so the last of enterocytes, I'm sort of obsessed about epithelium, so please bear with me. The last lineage we were really interested to look into was tuft cells. The reason being because tuft cells are this unique epithelial cell type with characteristic chemosensing ability. And what I mean by chemosensing is that these tuft cells are able to, for instance, during helminth infection, sense the parasites and translate these signals into something that is detectable by immune cells. So they're able to then secrete, for instance, Th2 response cytokines and activate ILC2 cells. So one discovery that Kylie actually made was that she saw that PLCG2 gene is very highly expressed by tuft cells, and PLCG2 is usually expressed by immune cells. And it's also a risk gene for inflammatory bowel diseases. So what we done next was we looked at what sort of receptors, we screened the receptors that could activate PLCG2 in tuft cells. And we observed that these Fc gamma receptors, 2A and 2B, were expressed by tuft cells as well, in addition to downstream molecules that are in this downstream PLCG2. And so Fc gamma receptors are have this function more in immune cells to sense IgG or immunoglobulin G. So this analysis sort of implicates tuft cells in being epithelial cells that are able to sense immunoglobulins and specifically IgG in the gut. And this was very interesting, especially our collaborators, Mena and her PhD student, Aaron, was very interested in this, and they've done a follow-up experiment where they looked at Fc gamma receptor expression in specifically in tuft cells in mice that are either healthy or DSS colitis mice, so basically inflammatory bowel disease mice. And they could see an increase of Fc gamma receptor in the tuft cells that are challenged with DSS, sort of suggesting that tuft cells might be important in response during inflammatory bowel diseases. Okay, so a little summary about the epithelial cells. So I talked about best 4 enterocytes and how we find distinct cells in different regions of the gut. I also talked about diversity of enteroendocrine cells and applying pseudotime analysis to better understand the differentiation of NPW positive enterochromaffin cells. And I also talked about tuft cells and looking at specific biosynthesis pathways to better understand the immune sensing or infer a new function in this cell type. Okay, so the next topic is about enteric nervous system. This is also something that we were quite excited about because we captured a lot of these enteric neurons in our data set. And so why are we interested in enteric nervous system? Well, actually, it's quite interesting because during development, these progenitors come from neural crest, and they colonize the intestinal tissues, usually through 2 different routes, either from the vagal end or to sacral end. And it's really interesting because these progenitors have to sort of maintain proliferation and differentiation, as well as the migration ability in order to reach the end of the whole intestine. And these progenitors then differentiate and create all these enteric neurons that are very important in regulating peristalsis and essential functions of the gut. And we also know that there are congenital disorders such as Hirschsprung's disease that are basically also called a ganglionic colon, which means that children born with this disease don't have innervation of the gut, of the colon, basically. And so we wanted to look at the expression of the genes associated with these rare disorders in our data set. So just to show you how this sort of put it in a context. So in our data set, we could see the appearance of these progenitors at around 6.1 post-conception weeks. And we wanted to visualize this within the developing gut. And we could see this ring of progenitors starting, well, kind of colonizing this layer, even at the time points where the epithelium doesn't quite even have the structure that we see in the adults. So it's really early on. And we could also, comparing small intestine versus colon, we could see that there's also a difference in how the cells appear, sort of looking, it looks more complete migration in the small intestine and less complete in the colon. When we looked at our single cell data, we could see that there are two different stages to this differentiation. So the first stage would be at 6 to 11 post-conception weeks, where the progenitors, both the glia and neurons, these blue progenitors, are highly cycling, and they predominantly differentiate through neuroblasts into neurons. And it basically shows two different branches of neurons, branch A and branch B, which is really similar to what has recently been observed in mouse model. And what we can also attribute is some functional attribute to these clusters. So you can see some of them, based on markers, seem more like excitatory motor neurons. Some of them are inhibitory motor neurons, interneurons, and IPANs. So there's quite a lot of diversity and differentiation that happens during the first trimester of development in the gut. And there's also a small population of glial cells that we observed at this time point. And so the second time point is actually quite different, strikingly different from the first one, where we could see primarily differentiation to glial populations, but still maintaining this differentiation towards neurons. Again, having branch A and branch B and different subclasses of neurons. But we also see these huge clusters of glial populations, as well as differentiating glia, sort of suggesting that gliogenesis actually follows neurogenesis. The other thing that we wanted to look at was whether these cells that we have, are vagal or sacral in origin. And for that, we use homebox genes. These are very well-known patterning genes that developmental biologists will know very well. And what we could see was that this glial population, especially the colonic glial population, seems to have lower expression of anterior Hox genes, but some remaining expression of posterior Hox genes, as well as specific expression of this TFAP2B gene, suggesting that this population probably is sacral in origin and actually colonizes from a different side. And we also could observe these cells in the mesentery, not within the gut, like I showed you in the first image, suggesting that they are probably also migrating in and giving rise to different innervation outside of the gut. Coming to the Hirschsprung's disease and looking at the expression of disease-associated genes, we curated a list that we were interested in and looked at its expression across these populations. But we wanted to preserve this difference between small and large intestines. So we looked at mean expression of each gene in both small in sort of counterparts within the same population, so counterparts from different regions. What was quite interesting here was that, for example, some genes like red was very highly upregulated in specifically branch A neurons. And the second sort of pattern that we saw was that some of the genes had slightly higher expression in the large intestinal progenitors compared to the small intestinal ones, possibly suggesting that perhaps either longer expression or sort of prolonged expression or higher expression of these genes is required for these progenitors to reach the end of the colon. We also saw some, interestingly, we saw quite broad expression across these lineages, but also that some of these genes are more upregulated in neuronal lineages, for instance, MAPK10, whereas others are more enriched in glial and progenitor populations, for example, SOX2 and SOX10. So there's a lot to explore here, but in addition to these genes, we also wanted to look at interactions between populations. So we know that there are these known interactions that influence the migration of these progenitors to the gut, and we wanted to see where do these ligands come from. Which cell populations secrete the ligands that attract these progenitors into the gut. And from this curated list, we could see that actually smooth muscle cells, as well as interstitial cells of Cajal, express very highly these ligands that are known to recruit progenitors into the gut. There was also some less expected populations, like these stromal populations, that also express these ligands. Yeah, so that was quite interesting observation as well. So in this part, I talked about neuronal populations that we see in the gut. And I told you how we identified 2 stages of glial and neuronal cell differentiation in the gut. How we can use this data to look at Hirschsprung disease-associated genes across many lineages, and the observations that we can find in this analysis. And then finally, how important it is to look at different lineages and how, what role they play in these biological processes. Okay, I have quite a lot of time still, which is great. So I'll move on then to the third part of my talk, which is going to be about immune cell organization. And this is, again, probably quite different from the rest, and a lot of information about the gut, but hopefully interesting. So the gut also, on top of this function of, absorbing nutrients, also has, is a barrier tissue and has a function in preserving immunity and living in harmony with all the microbiome that lives inside the gut. And the gut also has these structures, like local structures of immunity that are called gut-associated lymphoid tissues. And these highly organized structures have T, B cells, and all the different dendritic cells that help to organize these structures and activate the population in case of invading pathogens. So given that we have this developmental data set, we wanted to explore how do these structures form, and also whether these structures are important or relevant in disease. So what we know from model mice is that this process of lymphoid organization follows a three-step sort of process. And there are three main cell types that are involved in this process. So an example here is the organization of lymph nodes, actually, not specifically gall tissues, but in organization of lymph nodes, there are this population of lymphoid tissue initiator cells, or LTI cells. These are ILC3 type of cells, and they are characterized by expression of these surface receptors. They are recruited to these developing lymph nodes by the endothelial lymphoid tissue organizer cells. And these organizer cells secrete the ligands to attract these LTI cells. Once they are within these structures, then they are recruited further by the mesenchymal organizer cells, or MLTO cells, which then get activated and express a storm of cytokines in order to recruit the different T cells, B cells, and create these little hubs of local immunity in the gut. So within our data set, we wanted to look whether we actually do have MLTO cells or these activated mesenchymal cells. And amongst all this homogeneous data set, we could find a small population of cells that clustered closely with other immune-related fibroblasts, such as follicular dendritic cells and T reticular cells and others. And we could see that these cells actually increase in numbers, even though we didn't capture so many of them because these structures are quite sparse. But we do see these MLTO cells starting at 15 post-conception weeks and from both different regions of the gut, as well as the developing lymph nodes that we have sampled. And these activated cells express CCL21 and CCL19, suggesting that they are in activated form. What we also could see when we look at these H&Es, that there was also these little structures, endothelial structures, that resembled high endothelial venules for the entry of other immune cells to these tissues. So we could see that at 15 post-conception weeks, sort of the gut architecture is allowing or permitting the immune cells to colonize these gut tissues. And using small molecule FISH, we could also visualize the MLTO cells here in green, expressing CL13, as well as PROX1 positive lymphatic endothelial cells in proximity to these CXCR5 positive LTI cells. And so we further looked into the lymphatic endothelial compartment to sort of find whether we can identify this population that resembles ELTO cells in mice. And while sort of ELTO signals that have been described before were expressed across many different lymphatic endothelial populations, we could see one specific population, LEC2, that had expression of adhesion and recruitment molecules, which was very similar to the molecules that are expressed by this MLTO population. So we sort of had our candidate of ELTO cells as well within this data set. And so just a little schematic, now that we have our MLTO cells and we have our ELTO cells, do we also find the LTI population of cells that actually kickstart this whole process of organization? And so for that, we looked at within our T cell compartment, and you can see these are all the T cells from different ages of life. We can see both conventional and non-conventional T cells, naive T cells, and innate lineage like NK cells. But what really stood out was these populations here on top that were expressing LTI-like signature. And all of these populations had that signature that has been described in mice before. So this is a little representation of these populations that we find. And we could see that one of them had expression of the recruitment molecules that we would expect, but also reassembled ILC progenitors that have been previously observed in fetal liver. So they probably or possibly come into the gut from the liver. And then we saw another population that was ILC3, like clustered closely with adult ILC3s that are here in dark purple. And we inferred that there's possible maturation of this ILCP into this subset. The third population was actually NCR negative, and so differed from this population up here, and also expressed IL-17A, which is one of the molecules responsible for is characteristic of LTI cells. So it seems like instead of one population in humans, we sort of have three populations that possibly have this function of activating MLTO cells. We also visualize this using single molecule FISH. So we can see NCR positive ILC3s, we can see NCR negative ILC3s that express IL-17A, and also these ILCPs that were highly expressing this SCN1B molecule. And when we looked at the proximity of these cells to the MLTO cells, we could really see that the regions that had any MLTO cells had very low numbers of LTI cells, whereas in the regions where these green cells were observed, there's a storm of these PROC positive cells that are possibly recruited to this area, sort of suggesting that there is active recruitment of LTI cells. And so in order to better understand the sort of architecture of this process, we performed Visium spatial transcriptomics analysis on the fetal tissue. So this is fetal gut. And these different dots are Visium spatial dots with all the different transcripts that are captured within this region. And what we could see was that in this white box, we have actually captured a zone where this process happens. So I don't have it on the slide, but there was expression of CCL19 and 21, suggesting that the MLTO cells are there. And we have used the cell to location algorithm that has been also developed in SARS and Omer's group to map our populations to this tissue structure. And we could see that our LEC2 population mapped well into this area, as well as ILC, NCR positive ILC3. And in addition, we could see microfold cells that are characteristic of these lymphoid structures in adult starting to pop up in this area as well. And we also looked at what other cells are recruited to this area. So you can see all these different immune populations that we found are in the same zone as the MLTO cells and LEC2 cells and the ILC3 cells. Obviously, the next thing was, whether these structures are actually functioning during development. And for that, we looked at plasma cells. Again, the most important thing that happens within these lymphoid structures is the clonal expansion and activation of B cells that then differentiate into antibody secreting plasma cells. So we looked at the isotypes that are found in the fetal versus adult gut, and we could not see any class switch or clonally expanded plasma cells in development, suggesting that these structures need time to develop yet and need time to start functioning. and possibly it also requires perhaps microbiome and all these environmental factors in order to start functioning properly. Okay, so this is a little summary of this process. As I mentioned, there are 3 subsets of LTI cells that we identified, and we also find endothelial organizing cells and mesenchymal organizing cells in our data set. And these are all the signals that we infer from the single cell data to be important in this process. And so finally, looking how this model could be relevant in terms of inflammatory bowel diseases, we wanted to compare the populations that are found in Crohn's disease to the populations that we find in the fetal development. And what we could see using logistic regression was that ILC3s and Th17 cells were quite similar to the populations that we find during development. And there's also populations that highly resemble MLTO cells that are expanded in Crohn's disease. And so the next analysis that we did was we looked at Crohn's disease or ulcerative colitis associated genes. And we asked the question of which populations have the highest enrichment of Crohn's disease or UC associated genes. And these are all significantly enriched populations in these genes. And amongst them, we again can see the same LTI sort of populations that pop up. So this sort of suggests that these programs that are involved in development in order to recruit immune system into the gut might also again be activated during disease in active inflammation to recruit and retain immune populations at the site of inflammation to fight the invasion of pathogens. Okay, so in this part, I told you about the 3-cell model of gut-associated lymphoid tissue formation during the fetal development and how it might be relevant during the inflammatory bowel disease. And that gets me to my summary slide, actually. So I'm almost at the end. There's a lot of things that we explored in this data set, and that was really an interesting endeavor. All the data will be shortly available at gutcellatlas.org, and as well as in AriExpress and through Human Cell Atlas DBT platforms. So you'll be able to download, who's interested, and compare this data to maybe your data sets. And hopefully throughout my talk, there were some analysis inspiration that perhaps you can take on and do with your own data. And I'll finish with my acknowledgement slides. So there have been obviously a lot of people involved in this project, especially big thanks to Sarah and Kylie, who've been there from day one, and all the people from Sarah's group. We also have a lot of people from Sanger Pipelines involved in this project, and our collaborators, Maz, Mena, Omer, Matthias, Holm, Hamish, who's done the BCR analysis, and Matilda Katan, who is PLCG2 expert and has helped us to interpret the Tuft cell results. Kurosh, Roger Barker, and Stephen Lisgo for the access to tissues, to donors and their families, and to you for listening. Thank you very much. I'll take questions now.
Okay, so thank you, Rasa, for a very wonderful talk. It's absolutely amazing to see how the gut develops and the complexity of this organ throughout development, right? And disease as well. So you're tackling quite a bit of problems with your research, right?
Yeah, absolutely.
So first, we have a question from the audience. They would like to know if you could please emphasize on some of the software algorithms that use for clustering which ones maybe were more useful or which ones were more difficult to use, and what was the richness of using one over the other, right?
Absolutely.
And along the same line, we have another question by Benjamin Pizarro, so I'll just merge the two into one because they're very similar. If you could perhaps explain a little bit more in the technical aspects, maybe of the spatial transcriptomics analysis that you performed.
Yeah, so for all the analysis of this data, we used CanPy, and well, we used BBKNN for batch correction, and we found that works best because there's quite a lot of data and a lot of variables that we have to correct for. The biggest batch was coming from the developmental time points and different donors as well. So we've had to we corrected for actually each run in order to sort of regress this effect. Obviously, you know, that's not perfect, but given that these are human samples, and we also, this project sort of progressed as it went. We used different dissociation protocols that were improved as we go, and the tissues that we were, you know, collecting didn't always come at the same day and the same time, so that was a lot of variability there as well. But we found that BBKNN actually solved the challenge pretty well. So it mixed the cells pretty well in our case. So I would encourage people to try it out. With batch correction, obviously each data set will have quite different challenges. And so it's important to try different ones. I could point you to, there's a single cell course that we actually ran recently in EMBL, and there's code that is available in my GitHub account for different batch correction methods like Harmony if you're using R. So that course was based on R, and you can check that out. What other challenges we've found? Well, there was a challenge with doublet discovery. We used Scrublet. So that's a package for finding doublets, but I have to say that it wasn't finding all the doublets. And when you go to each lineage again, I would find again cell types that seem to be a mixture of different populations within the data set. And we've excluded these subsets because we didn't think that that's particularly informative in terms of what we were doing. But it's, again, a highly debated question. And I think, you know, depends what you want to do. The other package that we also applied in terms of quality control was Qcqc. So that's for removal of ambient RNA. It's a very helpful tool, although again, it's R, so it depends how big is your data set. Yeah. What else? What else can I tell you about the technical aspects?
It's a very broad question, right? So
it is a broad question, and there's a lot to do, and it's never enough, and more tools come up, and sort of, yeah.
And how was your experience with the analytical team, with the bioinformaticians, being from a wet lab background, right?
So I'm from a wet lab background, but I did analysis myself. So I sort of, during my PhD, I transitioned into the single cell analysis, and I was very lucky to have all the people in Sarah's group and who supported me in learning this analysis part. So yeah, but obviously there's still a lot of conversations that and it's not easy. I remember in the beginning, it wasn't really easy to understand, but the more you're involved in these conversations about what sort of issues you should be expecting in your data, the more you're like familiar with it, and the more okay it sounds.
Okay, so there's another question here by Lucas Pires Garcia. He was wondering, it's actually a very interesting question. You mentioned it, some of it, but how would you see the impact of the microbiome on the gut development? How are you correlating the microbiome profile with gut development? Because during development, the gut is practically sterile, right? There's very little exposure. So
we don't actually do that. So the study where we did where we correlated microbiome and single cells was in adults, so where we could actually sample microbiome. But during development, we don't actually do swapping, swaps of the tissue in order to get the microbiome, because I remember this one study which said that like the extraction reagents have more contamination than you would expect. So it's sort of pointless in a way because there will be contamination and you might be seeing some microbial species that actually are just coming from your reagents. So we didn't do that. Surely a very interesting question, and I think with different methods coming up, it's going to be answered. So
okay,
just to mention for the Visium data analysis, we use cell to location, and I'm gonna paste this to everyone.
excellent, on the chat, right?
Yeah, so panelists and attendees. So this is the package cell to location that we used to map the cells to the spatial zones.
Okay, perfect. We have a next question by Tiago Lubiana. He says, congratulations on the talk. It's amazing to see the real complexity of epithelial. How do you know that a population like the small intestine best 4 cells constitutes a new cell type? Did you have to do any type of registration of this new cell type as it is done for new species, or?
Well, we actually sort of didn't want to overhype it as being a new cell type. We sort of said, okay, this has been shown in other regions, and we also see it in small intestinal regions. So we just called it the same thing, and it didn't sort of raise a new cell type, hype. So we didn't have to go through registering new cell type or anything. I don't even know if people have to do that. I haven't had a chance to discover a new cell type yet, so yeah, didn't go through that. But yeah, completely agree. It's amazing how many different populations there are, and the way that we sort of identified it was, through clustering and looking at the genes and looking at the heat maps of how different these populations are based on differential gene expression. But what the package actually that might be useful for everyone that I sort of mentioned is Milo by Emma Dan. There's this bioarchive paper, and I'm sure you can find the scripts on GitHub. And this is differential abundance testing sort of method. So it helps to identify whether this population is, a new population or relates to other populations in your data.
Okay. Could you post the link to your GitHub as well, so that we can see the code for the other analysis that you mentioned before? That would be very nice.
Yeah, some of the analysis for this paper is not yet there. We've just submitted the bioarchive paper, so sorry about that. But I will post my GitHub where you can find the EMBL course actually that I mentioned.
Perfect. That would be really useful.
There you go.
Thank you so much. Now, I have a question now. Bear with me, because I have a medical background and I did my PhD on gastric cancer. So
wow.
I'm very, very interested in intestinal metaplasia, which is a phenomenon, right? By which
very interesting.
The stomach transforms, right? Transdifferentiates into first small intestine, that's called complete intestinal metaplasia, and then into the colon, or like colon-like structures, right? Which is incomplete intestinal metaplasia, each with increasing gastric cancer risk. So what's your take on, perhaps, I know that this isn't what you have done, but seeing how similar that process would be in the stomach to the differentiation of the gut during embryonic development, to see which programs are activated, which are kind of stopped during the transdifferentiation process.
This is definitely a very interesting question to explore, especially having the development and non-development samples. The thing that is missing is the metaplasia samples that you could then directly compare with the development. And I guess especially relevant would be stomach samples. We unfortunately don't have stomach in our data set, but
we have them. So if you're interested,
you have that. Oh, that would be awesome.
We didn't do single cell, but we have the samples. So
okay, I mean that's super interesting. We have, like, as far as I've heard, that there's this reactivation of sort of intestinal cells in stomach is super interesting. And as far as I understand, you know, in inflammation that happens more often, and then so the inflammatory bowel disease patients have a higher risk of developing cancers because this reactivation is sort of upregulated by inflammation. So there's definitely a lot of interesting questions to explore. I'm sorry, I haven't looked at the data myself, but I definitely think that there might be a developmental program that is reactivated similarly. But yeah, I haven't seen any papers on that, to be fair. So
okay,
very curious.
So we'll talk afterwards.
Yeah. Cool.
I think that's all the questions that we have for now. Anybody want to ask any additional questions to Rasa? Okay, so thank you once again.